Good afternoon, everyone, and welcome to our public meeting on evaluating real-world evidence from observational studies in regulatory decision-making, lessons learned from trial replication analyses. I'm Mark McClellan. On behalf of the Duke-Margolis Center for Health Policy, I'd like to welcome you to this event, which is being convened through a cooperative agreement with the Food and Drug Administration and Duke-Margolis. Today's meeting focuses on has a focus on a timely set of topics, as the current COVID-19 pandemic has highlighted the need for a better understanding of how real-world evidence can be used to inform clinical and regulatory decision-making. Observational or non-interventional studies that typically utilize real-world data, can broaden our understanding about the effectiveness of medical products. But questions remain about the credibility of observational studies using data collected for non-research purposes. I should say, too, there are other real-world applications that involve randomization, at least to some degree. That's not the main focus here today. Here, as part of its real-world evidence program, FDA is evaluating the potential role of observational studies in contributing to evidence on drug product effectiveness. So our virtual conference today and tomorrow is going to consider attributes of observational study design and study conduct that can help us better understand considerations for how to use the evidence from observational studies to inform regulatory decisions, and hopefully to also be useful for clinical decision making as well. There are a range of products underway that are seeking to replicate the results of randomized clinical trials that were conducted using randomized methods, with also careful attention to understanding data, so rigorous data collection. The goal here is to use observational studies with rigorous design as well, with the goal of understanding when evidence from these studies may be credible. Later today, we're going to hear from the lead investigators of a few of these replication efforts, as well as we're going to hear from other experts in the field. And this has been a very important area of applied scientific work to understand how real-world evidence methods and observational data analysis can fit in. So if you go to the next slide and go over the agenda for today, we're going to have 3 sessions today and then 2 tomorrow. Session 1 will feature a presentation on the target trial approach for designing observational studies. This can serve as an evaluation tool for considering what observational studies are appropriate and what is their ability to replicate the results of a clinical trial. Following that presentation, panelists are going to discuss the strengths and weaknesses of real-world evidence from observational studies, as illustrated by this approach. And then in session 3, our presenters are going to be joined by some additional panelists to identify some common themes, to react to the emerging insights, to suggest some ideas and issues about the path forward. So that's our topics for today. Tomorrow, we're going to cover some further steps related to key themes emerging from the replication efforts and observational studies, opportunities, limitations, and next steps going forward. Just a few reminders about this virtual meeting before we get started. You can visit the Duke-Margolis website for the materials from today's meeting, including a few relevant publications for the work that's being discussed today. At the end of session 1 and also at the end of session 3, we're also going to have some time to address any questions that you all want to submit or comments that you'd like to share for further discussion. If you have questions or comments for the panelists, you can send them via the Zoom Q&A function. That's at the bottom of your Zoom screen. Now, excuse me, we have a lot of registrants for this event, over 4,000 actually, so we may not be able to get to every question, but we are going to work through getting to as many or incorporating as many in the discussion as time allows. In addition, a recording of this workshop is going to be available on the Duke-Margolis website following the end of day 2 of this event. Because we are recording, we want to remind our speakers to please check that your camera and microphone are active before you start speaking. If you have any difficulty activating your microphone, our production partner can remotely activate or deactivate your microphone on your behalf. Presenters, as a reminder to you all, Valerie Parker, you'll see on the panelist list, is going to be advancing slides today. Please be sure to provide her with some kind of verbal cue, like next slide, when you're ready to move forward in your presentation. And in addition, Mira Gill, also part of our presenting group, will be timing the segments, and we'll be sending out reminders to the speakers and panelists through the Zoom chat function as well. For all of you, we encourage you to join this discussion on Twitter using the hashtag #TrialReplication. And we also invite you to follow the Duke Margolis Twitter account and join our ongoing discussion of these real-world evidence issues and other related issues online. Okay, so let's get going. Now I'm very pleased to turn to Jacqueline Corrigan-Karai, who's Director of the Office of Medical Policy within the Center for Drug Evaluation and Research at FDA. Jacqueline's going to provide some opening comments for our webinar today. Jacqueline, thank you very much for joining.
Thank you, Mark, and thank you everyone for joining us today, especially all our speakers and panelists for sharing their expertise. And while we'd love to be in person, I'm just really pleased to have so many people be able to join us virtually today. I want to extend a thank to the staff from Duke Margolis, in particular Adam Kresh, Trevin Locke, Joy Eckert, and Mira Gill, who've really worked tirelessly to prepare us for these 2 days of discussions, as well as perhaps you can show the slide of our steering committee that was kind enough to lend multiple hours to really advise on the agenda, so this will be a very productive meeting. And finally, to the outstanding team from FDA, including Dr. John Concato, Dr. Kim Pinto, Captain Biancarone, Commander Lopez, and Keila Holman. So in the past few years, we've just seen an explosion of interest in RWE, and probably an equal increase in the number of meetings. And, you know, we have to start everything with a definition. So for us, we just simply defined real-world evidence as clinical evidence about the usage and potential benefits or risk of a medical product derived from analysis of RWD. And for us, RWD are data related to patient health status and delivery of healthcare, routinely collected from a variety of sources, with routinely being a key part of the definition. We noted in the framework for our program that in addition to having data that's fit for the intended use, a key consideration is whether the trial or study design used to generate real-world evidence provides adequate scientific information to answer the regulatory question. We wrote that randomized trials can use real-world data both upfront and to improve efficiency, design, and recruitment, or to capture outcomes and generate real-world evidence. We spoke about the use of clinical practice data and registries as external controls for single-arm trials where randomization is not feasible. And the examples we provided were largely limited to circumstances that warranted appropriate regulatory flexibility in design, and where the real-world data really demonstrated a lack of treatment led to a predictable clinical decline in contrast to a significant effect of the therapeutic. And finally, we acknowledged there might be opportunities for purely non-interventional observational designs to inform the effectiveness of medical products, and that's really the focus today. We're excited to share results of the duplicate demonstration project on observational study designs and other demonstrations conducted to date, and to engage with our invited experts on what we've learned from this research. As we continue to explore the many ways that RWE can be leveraged to support our regulatory decisions, we are focused on taking a scientifically rigorous approach to the evaluation. However, we're not starting from scratch. We have over a decade of experience in the Sentinel initiative, including guidance on good pharmacovigilance practices and pharmacoepi assessment, that serves as a good starting point. In addition, we continue to innovate within Sentinel, including the use of new data and methods. A question we've been asked to really evaluate then is whether we can use these same methods that we've developed in the context of safety to evaluate the effectiveness of a product. We understand the interest in this area and also the challenges. As we have recently seen in COVID-19, RWD, when used appropriately, can provide knowledge about a new disease, its natural history, identify potential treatments for further study. However, the experience has also provided some cautionary lessons about the need for rigorous methods and transparency in designs and data. In our framework, we separated out data and design, but of course they're inextricably tied together. And the adequacy of the data is really the foundation that allows for an adequate design. We speak about real-world data, and the use of the term real-world data sometimes seems to imply that it's providing the real answers. And I think it's important to speak plainly regarding what data we are focusing on so we understand the potential strengths and challenges. Real-world data, in short, is clinical practice data. It overlaps with clinical trial data in both are clinical, both are real. But unlike clinical practice data, data collected for a clinical trial is captured in a prospectively planned, structured, and systematic manner to answer a specific question. The structure is what makes it fit for purpose because it's really designed around that scientific question. So what we're trying to evaluate is whether we can use clinical practice data, which does have some structure, and in particular, I think claims and certain drug dispensing data is much more structured, but in many ways it's predominantly unstructured and generally not systematically collected. It reflects an individual clinician's practice documentation. So in contrast to our trial data, we have to address the challenge of a lack of systematic collection. That means that specific measures and the timing of these measurements are not specified in advance. And while similar data may be collected, the timing and threshold for collecting such data, such as relevant clinical assessments, may be different. In addition, as we seek to follow patients over time, and they move in and out of the healthcare system based on employment and other non-health related factors. So our challenge is whether and how we can use these data to draw inferences about medical products, especially in designs where following up with those individual patients to capture the additional data we may want is not possible. As mentioned, we do rely on real-world evidence to inform safety for surveillance for products. Our reliance on non-interventional and observational RWE for safety is driven by certain considerations. Sometimes it's not feasible to get the answer through a clinical trial, or we need to explore a new signal more rapidly. In other cases, the magnitude of that signal that we see in an observational study may give us confidence that any potential bias from the lack of randomization is not likely to change our conclusions. Indeed, this parallels situations which we've used such data as external controls for effectiveness in rare diseases, including oncology. In addition, when you think about how a clinician prescribes a drug in clinical practice, if there's an emerging safety signal that's not really well known, it will not likely influence the clinical prescribing decision, and we're less likely to see the type of channeling we are concerned about that can lead to confounding, the issue that randomization is designed to address. Finally, I think we need to understand the regulator's perspective. Our regulatory threshold for including safety data in labeling and tolerance of uncertainty is a little bit different than the standard for reaching a conclusion about effectiveness. The focus of the regulator is not only understanding the effect of the drug, does it work, but also to understand the magnitude of that effect, to inform clinicians as they make trade-offs between benefits and risks and between products. The substantial evidence standard should be rigorous because once that determination is made that a drug is effective and those benefits outweigh its risks, it's unlikely that for most products, additional effectiveness studies for the same indication will be conducted. Indeed, our frequent reliance on two independent randomized trials, often with similar but not identical designs, recognizes that replication and consistency provide certainty to reach the substantial evidence standard. So it's not surprising that we think randomization is an important tool because it enables us to balance populations on clinical and other characteristics that we may not even understand. But this is not to say that randomization is infallible, as RCTs can be plagued by dropouts, missing data, post-randomization, and inadequate data quality. But it's a powerful tool, and no one's suggesting we're going to abandon randomized trials as the primary method for evidence generation for approvals, especially initial approvals. However, we need to explore whether there are opportunities for non-interventional analyses of clinical practice data to expand our understanding about the effectiveness of a drug that is on the market. Can such evidence be used to potentially expand the indication, perhaps address new populations, dosing, or longer-term outcomes? We have heard from a number of stakeholders that the development of new statistical methods, better understanding about design flaws that can lead us astray in non-interventional studies, and improvements in the quality of data, thereby providing more inputs for our statistical tools, opens up opportunities to answer a broader set of questions using these designs. However, it is not enough to postulate and pronounce. We are a science-based agency and we expect a science-based approach. So when FDA began its evaluation for effectiveness, in addition to supporting research,
All right, Jacqueline, I think we may have lost your sound feed there just now, about having a few issues before. Let me see if you're back.
sorry, yes.
Okay, there we go. There we go.
I lost my link. I don't know when you lost me completely.
Just a moment ago. So there are a few scratchy parts, but mostly fine. And I think if you pick up from just about, I don't know, half a minute ago, that should be great. Thank you.
Okay, sorry about that. I think I was starting to say how we had heard from stakeholders. If you heard that, forgive me, but it was more about that there's new methods and designs and that we can use these data for effectiveness. So when we began our evaluation of effectiveness, RWE, in addition to supporting research to understand data quality and the use of clinical practice data in regulatory clinical trials, we launched this duplicate project that you'll hear about today. The design of this research was to use non-interventional studies of clinical practice data to duplicate RCTs of regulatory interest. The goals were to better understand whether there are questions and study designs that could reach the same regulatory conclusions about these products. We did this largely using claims data, as you'll hear, and we've heard some critiques that with claims data, we are losing the richness of clinical practice data, and therefore this is a limited demonstration of what is feasible. While we understand that clinical notes and electronic health records contain much detail beyond claims, we have also seen the considerable challenges in consistently harnessing that information within and certainly across databases. Clinical claims data provides structure and potentially longitudinal insights into the medical course of care and have the benefit of representing actual prescriptions that are dispensed, more likely to have gotten to the hands rather than being prescribed. To date, claims data has been the backbone of FDA's Sentinel system to conduct non-interventional safety studies, and we have considerable experience here. The considerable experience with claims data, including algorithms to identify clinical events, made it feasible to do a number of replications expeditiously. Trials were selected if there was sufficient clinical practice data on the use of the drugs, and the selection of the population and identification of the endpoints were feasible. The point really was to focus on the methods by picking trials for which claims data were potentially already fit for use. We have also heard that we're somewhat comparing apples and oranges. In clinical trials, there are controls and procedures that lead to high adherence and more homogeneous patient populations that cannot be duplicated in clinical practice. Again, we acknowledge that there are differences between the use of medical products in clinical practice and controlled trials, adherence perhaps being most notable. However, if a robust, well-controlled RCT has established effectiveness, it's reasonable to treat that as the gold standard or reference standard against which to benchmark, and then we can understand if directionally there is comparability, but perhaps differences in the effect size that might be due to differences in clinical settings. In addition, as you'll hear more about today, our goal was as to closely as possible duplicate the design of a well-controlled conducted RCT, including the study populations. In addition, we have emphasized best practices for conducting non-interventional studies and transparency, recognizing that these types of studies open the door to multiple data analyses, the so-called Mulligan problem. Our colleagues from Brigham and Women's will share not only the results, but the steps taken to harness best practices and transparency, including through registration on ct.gov. Finally, whenever you have a number associated with an activity, our natural tendency is to generate a scorecard. The point of this activity was not to declare the demonstration project positive if it crossed the threshold and negative if it did not. We actually think that any failures are just as informative, if not more so, than the successes to understanding the feasibility of this approach. In addition, what is presented is what was duplicated, and certainly we also need to understand the broader universe of trials that we selected as potentially duplicatable and then stopped. So our goal today is understanding what this demonstration projects and others that will be presented today tell us about the use of non-interventional studies for informing effectiveness. What lessons have we learned? What questions remain? And what are the appropriate next steps as we continue to evaluate these studies? So I look forward to a thought-provoking 2 days and turn it back to you, Mark. Thank you.
Great. Thank you very much, Jacqueline, for that overview of what we're going to be covering over the next couple of days and how it fits with FDA's broader approach and strategy related to real-world evidence. Just before we move on to the first session, a number of questions came up about some logistical details. So there were not slides for Dr. Corrigan-Karai's presentation. Is meant to be a synthetic overview. There are going to be plenty of slides coming up for the specific topics that we'll be covering. And for those of you who are not familiar with the FDA's overall strategy on addressing real-world evidence questions for regulatory purposes, FDA's published a framework on the use of real-world evidence that highlights the key pillars of whether the data are fit for purpose, or rigorous enough or appropriate enough for use. And we're going to discuss that today in the context of some of the claims data that were used in these replication studies. Second, whether the methods are fit for purpose. Remember, these are observational studies, so could involve potential sources of bias, and that's a big objective. And we'll talk about this as well in the next session around the notion of a target trial design, big objective of this work as well. And third, as you get into specific applications, was the mechanism for using the data and the methods appropriate to the question at hand. So some frameworks available from FDA. Also, if you go to our website at Duke Margolis, there are a lot of links to additional resources as well. So right now, I'd like to start off down the filling in the overview that Dr. Corrigan-Karai provided as we go to the first of our 3 sessions today on principles for using rigorous observational study designs to replicate the results of clinical trials. We're going to hear from Miguel Hernan, who's the Colocotrones Professor of Biostatistics and Epidemiology at the Harvard T.H. Chan School of Public Health. He's going to talk about the target trial approach to trial emulation. Following his presentation, we have panelists, 4 panelists who will discuss some of the strengths and weaknesses of real-world evidence from observational studies, really the issues to look out for in this overall framework that we've started to discuss, including considerations for suitable clinical context and appropriate use. Our panelists for this session include Adrian Hernandez, who's the Executive Director of the Duke Clinical Research Institute and also Vice Dean and Professor of Medicine in the Division of Cardiology at the Duke University School of Medicine. Nandita Mitra, who is a professor of biostatistics and vice chair of faculty professional development in the Department of Biostatistics, Epidemiology, and Informatics at the University of Pennsylvania. Jennifer Graff, who is the National Pharmaceutical Council's Vice President of Policy Research, and Gerald Delpan, who serves as the Director of the Office of Surveillance and Epidemiology in FDA's Center for Drug Evaluation and Research. So Miguel, I'd like to turn to you now, and I think you have some slides to present as well.
Thank you very much, Mark. So I'm going to be talking about causal inference from observational data, which is a different way of referring to target trial emulation. And we want to make causal inferences because we want to learn what works and what doesn't work. And the way that we learn what works, as we all know, is we conduct a randomized experiment. That's the best possible way of doing it. For any question that we have about comparative effectiveness or comparative safety, there is a randomized trial in principle that would answer that question. Of course, as we all know, we don't always have randomized trials, and the previous speaker has already explained a few of the reasons why. They may be expensive, not ethical, not practical, or simply not timely. We will see some examples in this talk in which we will eventually have a randomized trial, but decisions have to be made before we have the randomized trial. And when that happens, what we do is to analyze observational data. And something that I want to make very clear is that we analyze observational data or real-world data because we cannot conduct a randomized trial in the population that we want with the treatment strategies that we want. Otherwise, we wouldn't do it. So observational analyses are not our preferred choice. We just think for each question, for each causal question that we have about comparative effectiveness or comparative safety, we can think of a hypothetical randomized trial that we would prefer over an observational study. And that hypothetical randomized trial that would answer our question, if it were possible, is what is really the target of our inference. So we refer to it as the target trial. The target trial is the hypothetical randomized trial that we would like to conduct to answer our causal question, to learn what works. So we can think of causal inference from observational data as an attempt to emulate some target trial that we have in mind. And we can take this analogy further. If we are doing an analysis of observational data and we don't know, we cannot articulate what the target trial is, then probably we don't know what we're doing. This concept of a target trial is not new. It's something that has been central to causal inference theory for a long time. The first mention that we have found was by Dorn at NIH in the 50s. But Cochrane, Rubin, Feinstein, David in Cambridge, they all have used this concept in some way or another, even if they didn't use the label target trial. But they all also were working mostly with simple treatments, simple exposures that happen at one time only. And we have to wait until the 80s when Jamie Robbins made this concept of a target trial general, so that we can apply it to treatment strategies that are sustained over time, to time-varying treatments. So if we think about causal inference from observational data as an attempt to emulate the target trial of interest, then we can organize causal inference in two steps. The first step is that we have to ask the causal question, and the second step is that we have to answer the causal question. And a lot of the emphasis among those of us who teach methods and would work in this area has been step number two. But actually, step number one is even more important, because if we don't ask a good causal question, we cannot answer it. And how do we ask a causal question? Well, we have to specify the protocol of the target trial. And then once we know what which the target trial is, then we can conduct it if we can, or use observational data, use real world data to explicitly emulate the target trial. And explicitly is really the key word here. So what I'm saying is that if we are doing an analysis of observational data, the first thing that we have to do is to design a randomized trial. And then when we design this trial, we write a protocol that has the main elements of the protocols of all randomized trials. Who is the population? What are the eligibility criteria? The treatment strategies, how we're going to assign treatment, the start and the end of follow-up, the outcomes, the causal contrast, or as is now known, the causal estimate, and the analysis plan. And then once we know this for the target trial, we know what our causal question is. The next step is to emulate each of these components explicitly using the observational data, if we can. So when we don't do this, when we don't know the target trial before we start analyzing the observational data, it's really like shooting without a target. We have no idea whether we are going to get it right or not, because we don't even know what our question is. And this is a problem that has been exacerbated in practice and in the medical literature because of the reluctance by many journals to explicitly state what the causal goal of an observational study is. We see a lot of studies that are about associations, as opposed to saying, look, the goal of this observational study is to estimate this causal effect, which we are doing through this association. but because we want to estimate this particular causal effect, this is how we are doing the analysis. In the absence of being explicit about what the causal target is, a lot of the methods that are published in observational studies are very hard to evaluate. So why is this important? Why do we need to explicitly emulate a target trial for causal inference when we're using observational data? And the reason is simple, is because when we don't do that, we have a higher probability of getting a biased estimate. And I'm going to illustrate this with 3 examples that we've been working on over the last decade or more. And the first example, oh, sorry, some very important point before I move there. When we are using observational data to emulate a target trial, the target trial cannot possibly be a placebo-controlled trial, or a trial with double-blinded design, or a trial in which there is a very intense monitoring of patients every day. That doesn't happen in the real world. So we cannot emulate target trials like that. The target trial that we can emulate with observational data will rarely be a phase 3 trial. It will be the trial that you do in the case of drugs in the post-market setting. Now, the first example is hormone therapy and heart disease. So very quickly, because I'm sure that many of you have heard this example before, I still like to use it because it encapsulates a lot of the problems with analysis of observational data. So this is a situation in which observational studies for a decade or 2 had been had found that women using hormone therapy in menopause, after menopause, had a lower risk of heart disease than women not using hormone therapy. And when a randomized trial was finally conducted, it found apparently the opposite. It found that women assigned to hormone therapy had a higher risk of coronary heart disease than women assigned to placebo. And if you think about what happened here, is that the observational analysis wasn't even trying to emulate a target trial, because what the randomized trial did when it was conducted was to compare women assigned to hormone therapy versus women assigned to no hormone therapy. Women assigned to hormone therapy were the women that almost all of them, they took at least 1 pill of hormone therapy. So the trial was comparing initiators, incidence users or new users of hormone therapy versus non-initiators of hormone therapy. In the trial, if you look at the data from the trial, what you see is that the hazard ratio over time starts to go down. It starts at 1.8 in the first year, 80% increased risk of heart disease. Then it goes to 1.5 after 2 years. Then it keeps going down. And after year 5, the hazard ratio is 0.7. It's 0.7, of course, not because hormone therapy becomes beneficial after 5 years, but because there is a depletion of susceptibles, which is why when we give the hazard ratio from a randomized trial, we use this average over all possible, over the entire follow-up. We don't delete the first 5 years of follow-up and tell you that the hazard ratio after 5 years was 0.7, because even though the hazard ratio was 0.7, the cumulative incidence, the risk of heart disease was always higher in the hormone therapy group. What the observational studies did was, for all practical purposes, they deleted the first few years of follow-up because they compare current users versus never users. And current users, the women who are current users now, are the women who have survived using hormone therapy for some period of time, and therefore are more likely to be the least susceptible women that, despite being using hormone therapy, have not developed heart disease. So once the observational data here was reanalyzed exactly in the same way that the data from the randomized trial was analyzed, the observational studies also found an increased risk of hormone therapy. So this wasn't a problem with the observational data. It was a problem with how the observational data were being used. you get very compatible answers between the randomized trial and the observational studies, both when you do the right analysis and when you do the wrong analysis. Now, let me give you a more recent example. There are a large number of studies, of observational studies, that have found an association between statin use and lower risk of cancer. Sometimes 50, 65% lower risk of cancer. Now, these observational studies, which are published in the best medical journals that you can think of, they are wrong. And we know that they're wrong because there are meta-analysis of randomized trials of statins that have found not even a hint of an effect of statins on risk of cancer. But also we know that these observational studies cannot be so wrong because of confounding, because of lack of randomization, because statins are not prescribed with an eye on cancer risk. We don't use risk factors for cancer to prescribe statins. We use risk factors for coronary heart disease to prescribe statins. So it is very unlikely that this association that doesn't have a causal interpretation in observational studies is the result of confounding, is the result of lack of randomization. So we explored this issue and we emulated the target trial of statins and cancer using electronic health records. In this case, we were working with a Caliber resource from the UK. And the first thing that we did, of course, was to describe the protocol of a target trial. So very quickly, and I'm not going to go over this, we just went over the eligibility criteria. These are people with no history of cancer, with no contraindications to statins, etc. That would be assigned to either initiation of statin therapy or no initiation of statin therapy, and then followed until a diagnosis of cancer, death, or the end of follow-up. And we will look at both total cancer and site-specific cancers. So after doing this, and I'm going to show you only the results of the observational analog of an intention to treat analysis, there was nothing. The hazard ratio was 1, essentially, or very close to 1, both for total cancer and for every site-specific cancer that we looked at. The cancer-free curve was also were overlapping between the statin group and the non-statin group. But again, this is very different from what many previous observational studies have found. Why? Well, again, the reason is that many observational studies did not even attempt to emulate a target trial. And I'm going to give you one example. There was a study published that found an odds ratio of lung cancer for long-term users of statins versus non-users of 0.23. And I'm not kidding, this is published in a serious medical journal. So someone is saying that statins lower the risk of cancer by 80%, of lung cancer by 80%. And that's published without raising a lot of eyebrows. So what is going on here? Well, again, this is an example in which prevalent users were used at baseline, like in the hormone therapy example. And it's also an example in which something even worse is being done. It's that data that are not available at baseline at the time of assignment to treatment are being used to assign people to a particular treatment group. In this case, you only assign people to long-term users of statins for more than 4 years, if you observe that they have been using statins for more than 4 years. But you never do that in a target trial. You never do that in a randomized trial. You don't assign people to different groups of treatment based on what they have done in the future. You assign them to different groups of treatment based on what they do at baseline. When we did this with our own data, we also found a hazard ratio around 0.2 or 0.3. But again, the problem here wasn't the observational data, was how the data were being used. If you want to look at long-term users, long-term use versus non-use, there are specific methods to do that are not, definitely not, put in those who have been using the treatment for more than 4 years in one group and non-users in the other group. And this, everything that we are saying affects also to case control studies. In fact, many of these studies of statins and cancer are case control studies. And there is one that was published in the New England Journal of Medicine that found also an incredibly strong inverse association between statins and cancer. We were looking at that in more detail, and you have the reference there, and you can see why that case control study was finding that association. It wasn't emulating a target trial. If we have case control data, we will do the same thing. First, we have to specify the protocol of a target trial. Then we have to define what is the cohort study that we will use to emulate the target trial, and then sample the cases and the controls from that cohort. And that will be our case control study. The key point I like to make in that in these examples, the problem with the observational studies was not lack of randomization. The problem was not confounding. And this is something that we keep finding over and over. A lot of the criticisms of observational analysis are that they are not good because they are observational, because there is no randomized assignment of the treatment. But what we keep finding is that a lot of observational studies that are published are incorrect for reasons that have nothing to do with lack of randomization. They have much more fundamental problems, which in the last two examples that I show you is essentially it's a failure to choose time zero and to handle time zero in the right way, right? If you think about what time zero is in a randomized trial, is the time for each person when the person meets the eligibility criteria, when a treatment strategy is assigned to that person, and when we start to count if there is an outcome. That's obviously what time zero is in a randomized trial, in a target trial. Therefore, if we are doing observational analysis, we have to use the same definition of time zero. Otherwise, we run into problems like what I just described for hormone therapy or for statins and cancer. In those studies, there was a misalignment of the eligibility criteria and the treatment assignment, which leads to selection bias or immortal time bias. Anytime that here the dotted vertical line is time zero, every time that the eligibility E and the assignment to treatment A are not aligned at time zero, we are at high risk of bias. The second example in this graph is the hormone therapy example, in which at time zero, the eligibility criteria were applied, E, but the assignment to treatment had happened before. So because women had been using hormone therapy for a long time before, the hormone therapy itself had affected the possibility of being eligible for the trial, and that creates bias. That would never happen in a true trial. It happens in many observational analyses. And the last of the four examples that I show you there is the example of the immortal time bias that I was describing for the statins and cancer example. At time zero, we apply the eligibility criteria, but we assign people to treatment based on the treatment that they took after baseline. So when we think about emulating a target trial, we have to think always of two components. One is the randomized assignment, and we emulate that adjusting for compounding. Adjustment for compounding is what we do when we want to emulate randomization. And we may fail if we don't adjust for all the confounders, and that's of course a big problem of observational studies. But an even bigger problem in practice is the specification of time zero. We keep seeing examples of observational studies that are wrong because time zero is not because we don't have a synchronization of eligibility and assignment at time zero. So if we want to improve observational data and target trial emulation, the low-hanging fruit is thinking more about how to choose time zero in the right way. Now, this doesn't mean that it's okay to use observational data once we fix the time zero, because of course we still have the problem that it is not randomized and there may be confounding, and that's a big problem that we always have to keep in mind. And there are many examples in which observational data we are finding cannot be used to emulate the target trial. We have tried to use claims data to estimate the effect of preventive interventions on mortality. It's impossible. It doesn't matter which fancy method you use, machine learning, anything. It's just not possible. And also there are some treatments in which the confounding by indication is so extreme. Everyone who has certain indication takes the treatment that you cannot use observational data either. So there are limits. But so this business of using observational data to emulate target trials was really not very useful in itself, right? Because if we know the answer from the trials, then why spend time trying to replicate them? We do that because by replicating trials that already exist, we understand better under which circumstances target trial emulation is possible. And as the previous speaker said, it's very important to publish, not only when we succeed, but also when we don't succeed. It's very important to publish. We have tried to emulate this target trial using this particular database, and it wasn't possible. So everybody knows. It's actually very hard to publish those things. I can speak from experience here. Most editors don't want to publish failures, which creates problems. So let me finish with one example for the COVID era, the example of tocilizumab. Okay, so this is an antibody against interleukin-6 receptor, and it was observed already in February in China that people with high levels of IL-6 have an increased risk of death when they had COVID. So people started to use tocilizumab to treat serious cases of COVID-19, but there were no randomized trials. So we are in the spring of 2020, there are no treatments, no known treatments that work. This is a possibility, people use it. So we conducted an observational study using about 4,000 individuals in the ICUs in almost 70 hospitals in the US to find to try to estimate the effect of tocilizumab on mortality. And as you can see, the paper was published, and the first word of the paper is association. That's journal policy, even though the whole paper is about target trials and causal effects. So this is what this observational study found. It found a lower mortality in the TOSI group compared with the non-TOSI group. This is after applying inverse probability weighting and other methods, and doing something reasonable to choose time zero without risk of immortal time bias. There was an almost a difference of almost 10 percentage points. The hazard ratio was 0.7, and the hazard ratio was even farther from 1 for people who had been admitted to the ICU recently. So apparently, a very strong effect of tocilizumab on mortality at a time when there are no known treatments. So what happened? Well, it wasn't taken very seriously. This research was finished in May, was around several journals until it was published. And I'm not saying this to whine about not having your paper accepted. I really want to make a point, because what I'm showing there in quotes is what the editors of the journals were saying. And this is important because it tells us how people see observational studies, how people say the potential of emulating a target trial using observational data. The first editor says, I'm sorry to say it was not accepted for publication. This was an editorial decision. The decision was to wait for actual trials, essentially saying we don't trust observational data. The second editor says there was concern that there was a high risk of residual confounding, but that is not supported by any argument about why there could be a high risk of residual confounding after adjusting for everything. Even if there had been a lot of residual confounding, it was in the opposite direction. It was making tocilizumab patients look worse. And follows, none of the randomized clinical trials that are beginning to report have found such an effect, which is not true. The trials were tiny. They had 60 people, 70 people in each arm. The confidence interval was so wide at that time in those trials that nothing could really be said. So a few months later, this is published. After it is published, it wasn't taken seriously by people who write the guidelines. In fact, tocilizumab has been, the use of toci has not been recommended during much of the pandemic, except maybe in the UK now. And if you read the treatment guidelines for the National Institute of Health, in their brief summary of evidence, as of last week, they didn't even mention the observational studies. So when the large trials were published last month and last week, they're not published, but the results are known now. They found the same thing as the observational study had found in May. And this is not surprising because this is precisely the poster child for when target trial emulation can work. You have a very large causal effect. We have data with rich information on confounders. The residual confounding goes in a conservative direction, and there is a sound design of data analysis and handling of time zero. So if it doesn't work for tocilizumab, if we cannot trust target trial emulation for something like toci, I don't know if there is hope that people will ever trust target trial emulation for anything else. In this particular case, journal editors, guideline writers, regulators, they ignored the observational analysis. But they did so without a criticism. They just said they were observational and therefore we cannot trust them. And this happened during a public health emergency when there was no effective treatments. So this was the perfect setting for observational studies like this to be taken seriously, and it didn't happen. That's how biased we are against observational studies. Not even in this situation we use them. And of course, this is not very surprising. It's the result of so many bad observational studies that have been published over the decades. So as I said, the target trial concept leads to a very simple two-step algorithm. Ask the question first, then answer the question, specify the target trial, try to emulate. And my last slide here is really a message that I like to convey, that every time someone presents observational estimates, when they're trying to estimate causal effect, just ask them, what is the target trial? Because if they don't know, you can help them. And in fact, sometimes you can find that there is no target trial that can be mapped into the analysis that they are doing, which is great. Then they can start over. And the really important point is that only after we know the question, only after we know what the target trial is, we can evaluate the methods that are used to obtain the answer. Thank you.
Thank you, Miguel. Next, we'll hear from Adrian Hernandez. Adrian?
Okay, all right, Miguel, a lot there to digest here. And so, and I think the COVID example kind of brings out a lot for us to consider because there has been the so-called good, bad, and ugly in our COVID experience in terms of observational studies versus what we see for clinical trials. And I'd say like the main thing that we're trying to solve is that we're really good about advanced hindsight. So looking backwards to say, hey, this observational study replicated a trial for good reasons, or it didn't, and the reasons why it wasn't replicated was for A, B, and C, to getting to something that's more advanced foresight. How can we actually plan out, when an observational method or study or studies can actually inform us in terms of either clinical decision making or enable regulatory decision making. And so I think there's a framework here for us to consider as we go forward, as we'll hear more through the day and tomorrow about examples where people have taken robust approaches in a rigorous fashion to replicate clinical trials, as well as share when they haven't been replicated. And so if you think about it this way, is that if there is a medical product on the market or gets on the market after a robust clinical development program through phase 3, where it's very clear that we understand the benefits of risk, then, and if you can replicate that clinical trial, then perhaps that gives license to doing other things with observational studies that couldn't be done in randomized trials. For example, there may be populations that have been understudied, for which actually it would be difficult to do a randomized clinical trial, either because of lack of individual equipoise, even if there's community equipoise, or because of the cost, or because of where it is on the life cycle, if it's later in the life cycle. And the other thing is that for comparative effectiveness, it certainly seems right for us to understand that with new methods. If we did have that kind of ability where we were replicating, say, large phase 3 programs, and then we're aiming to extend information in the label or for clinical decision making, then that could be appropriately marked. That may not be necessarily the same as a phase 3 trial for an indication, but certainly can provide data for decision making, both at the clinical level, guideline level, and even perhaps the regulatory level, and could do so in a structured way. And so let me just give kind of 2 examples here of kind of potential use cases. We've had drugs that have been recently approved, or not necessarily recently approved, but new agents over the last several years in the field of diabetes focus on hyperglycemia. And certainly there's been development of robust data in terms of the benefits and risk among patients with diabetes. But now we've seen increasing evidence in terms of its use across other ancillary conditions, heart conditions, renal conditions, and others. And so you can imagine as a life cycle develops, how would you expand our knowledge base through observational methods if you start off being able to replicate the early phase 3 programs, and in an ongoing way, the safety surveillance didn't show any safety concerns through usual practice. But you also have to be careful, as Miguel noted. If you don't have robust methods, you could unfortunately lead to wrong conclusions. I'd say the COVID-19 is another use case where we've seen both. So you can't get too far before mentioning hydroxychloroquine or convalescent plasma, where observational studies haven't been replicated in terms of clinical trials. And so it does show that there is this need to have a robust structure or framework where you start with a replication or emulation of something that's so-called known with the randomized trial, and then give license to be able to expand information that could be part of a label even for an indication or expanded indication, or more information about a population. In heart failure, as an example, there have been studies that haven't been done in the US for which we don't actually know the effectiveness or efficacy, perhaps in randomized data for populations of interest, especially for Blacks or people of color that have not been represented in those trials. But it would be hard to do a trial in that setting these days that's really a social construct as opposed to a biological construct here. So with that, I think we have some ways where we can move forward thinking about emulation and then expanding information based on the needs for a population of interest or expanded indication. So I'll turn it back over to the next panel.
Thank you, Adrian. Now I'll turn it over to Nandita Mitra. Nandita.
Hi, thanks very much. Thanks to Dr. Hernan for an excellent presentation, and to Dr. Hernandez for his very thoughtful comments. I'd like to really kind of share my experience as a statistician who collaborates with clinicians who are primarily interested in observational studies. And so when I meet with a clinical collaborator who wants to, say, estimate the effect of a particular treatment from observational data, say SEER Medicare, because I work primarily in oncology, they're often aware that they should have an observational study protocol similar to that in a clinical trial. And this is already, somewhat new thinking and is highly to be highly commended. And much of the credit goes to Strobe, the strengthening the reporting of observational studies in epidemiology checklist and guidelines, and more recently Spiros, where they give guidance on observational study protocol reporting. And researchers mostly agree that not having a detailed protocol, at least written, if not registered, prior to data collection, limits the ability to assess intentional or unintentional bias. And a detailed study protocol is also essential for replicability. But most of my clinical collaborators that I work with haven't thought carefully about framing their studies design in the context of emulating a target trial. And oftentimes when researchers come to me to talk about their observational data, they jump immediately to the statistical approach that they'd like to use. So for example, collaborators are often really excited about wanting to propose a propensity score analysis or an instrumental variable analysis because they have just heard about it, learned about it, read about it in another clinical paper. I've also seen a recent uptake in the number of collaborators who'd like to do a difference in differences analysis. Or collaborators may want to use something that's familiar to them. So they may say, I want to use a Cox model because they're familiar with it and they want to use something that they're familiar with. But it's basically defining a statistical approach before they've actually thought about what they actually want to estimate, the target estimate. But by defining a Cox model, you're automatically kind of backing into determining that you have to then estimate a hazard ratio. Is this really what they want, or are they just backing into it because of a statistical approach that they'd like to use? So I try to emphasize the importance of not specifying a statistical approach first and then backing into the target estimate. It's really critical, as Miguel mentioned, to think about the design of the study. We need to carefully think about, as Miguel mentioned, the eligibility criteria, the treatment strategies, the assignment procedures, the follow-up period, the outcome, and the contrast of interest, the causal contrast of interest. And then, and only then, should we jump to thinking about the analysis plan. So once we have a good idea of what we want to estimate, we can then think carefully about the estimation procedure. So then we can get fancy with our methods. If there's time-dependent confounding, maybe we'll use G computation. Or if the outcome is highly skewed and there's zero inflation, maybe we want to use something really flexible, like non-parametric Bayesian approaches, like Dirichlet processes. But again, all of these approaches that seem really sexy need to come later, after we've thought carefully about the study design and the estimate. So to wrap up, my take-home message really to my collaborators is, when you have observational data and you want to conduct an observational study, think very carefully about the design and the target estimate so that you are emulating a target trial, a very new way of thinking amongst most of my collaborators, and then tailor the statistical approach accordingly. And so in other words, if you allow me a dorky statistician pun, don't put the BART before the horse. Thank you.
Great. So I believe that I'm up next, and so I'll just jump in. And I want to thank the team at Duke and Dr. Hernan and FDA's interest in this. I think all of us that are on the call today believe that we want observational studies to be taken seriously, but we want to understand what matters. And so I'd like to offer up 3 points on what matters. First, words matter. 2, what questions you focus on matter. And 3, the methods matter. And I suspect that many of us on the call today are thinking about what methods matter. So why do we focus on these things? Well, I spend most of my time thinking about what type of evidence we can try to use to answer questions for population health decisions and reimbursement decisions.
Jennifer, I think we may have lost your feed.
I have to make about
Sorry, Jennifer, we lost you for just a second there. You may want to repeat the last little bit.
Sure. So the last part is methods matter. And I think for anyone that's on today's call, you likely are very focused on methods. As I was saying, why do words matter? Words matter because many of us are thinking not only about what the FDA decisions are, but what are the decisions for patients that are needing care today and how we pay for care decisions. That's where I spend most of my time. And for many of those in the audience, there's a vast loss of information that they need evidence to better understand what works for patients when they're trying to make decisions, and the patients in the office or the payments in the office. So one of the things that I'm excited about hearing today is the change as we've been talking about things. Are we talking about emulating target trials? Are we talking about replicating clinical trials, or are we talking about duplicating those trials? As we think about benchmarking, that's one of the words I heard Dr. Hernandez describe, that really seems to me to resonate with what we're looking at. We're looking for a few performance indicators to help us understand whether observational studies can help meet the performance that we know and can trust from randomized controlled trials. It can't be every endpoint. It can't be a chinning bar. And it's something that's likely to change over time. So as the methods and data get better, it's not a static measure. And so understanding what that means to emulate a trial is really important, not only for those on today's call, but also for those in the broader context that may not have thought as carefully about regulatory replication, statistical replication, and all of the permutations that can occur. So the second part of what matters are the questions. And there are numerous examples that we're hearing later on today about what studies you can actually emulate. A few years ago, we did some work with Teresa Gibson using market scan data to try to emulate similar and high-profile comparative effectiveness trials. And I think there's two key things that we need to think about as we try to figure out what we can emulate or what we want to emulate. It's pretty straightforward if you can't measure the outcomes. It's not data that's fit for purpose. However, if you try to emulate a target trial using all of the inclusion and exclusion criteria, you'll likely end up with less than 1%, as we found in our attempt. It's only when you start to try to figure out, well, which inclusion and exclusion criteria are really critical? Which ones could likely bias things and affect depletion of susceptibles or have time-related biases due to events occurring both before and after the time zero. And so really figuring out what studies we could realistically emulate is quite important, because otherwise we're likely to say, oh, can we trust it? Should we do it? And do we want to trust those results? Those are important issues. The third issue that we want to think about are the methods. And the beauty of today's work is really focused on high quality methods. Each one of the future presenters is going to show some really robust data, methods, etc. But we know that for observational studies out in the world, published, etc., that we have a large opportunity to improve the quality of the studies, just as Miguel mentioned around time zero issues. Some work we've recently done with Jesse Franklin and Kate Bykov looked at a systematic review of 75 studies, cardiovascular, diabetes, osteoporosis, a lot of where you see observational studies. And we looked at the presence of methodological flaws and 4 of the major flaws. Not surprisingly, over 50% of that sample had a time-related bias issue. And most of the studies had at least one issue. So I mentioned before that performance and benchmarks aren't static. So as the opening remarks mentioned, well, why not just use electronic health records, or perhaps use more recent studies versus studies from 10 years ago? Unfortunately, what we found is using EHR linked data didn't matter. More recently published studies didn't matter. It's things such as using active new user control designs and active controls that really help to or were associated with far fewer flaws. So I think for those of us on the call, we should think about what are the methods that we're using. And as Dr. Hernan mentioned, there's many things within our control, not just the randomization, that can help improve whether observational studies can be trusted. So if we want them to be taken seriously, we need to be very careful with the words that we use as we describe the observational studies and whether they meet the bar for emulation or whether we're talking duplication and replication. We need to be very careful about what studies we're trying to replicate, and also which studies we even think RWE is appropriate for decision making. And third, we as a professional society, as professionals conducting real world evidence, we have a lot of room to improve the methods. And it's only by doing this that we can ensure that observational studies can be taken seriously, not only for regulatory decisions, but for all those decisions that touch each one of our lives as we determine what treatments we should be taking and or which treatments should be paid for. Thank you.
Thanks very much, Jennifer. And next we're going to go to Gerald Delpan.
Okay, so thank you, Mark, for organizing this, and to my colleagues at FDA for organizing this. And thank you to Miguel for such a great presentation, and to the panelists for their comments. It's hard to add a whole lot afterward, but let me start by saying something obvious. One of them is this is not easy. There really are a lot of considerations that you have to make when designing an observational study. And I like the term that Miguel used, the self-inflicted wounds. These are things that presumably could be corrected if people thought more about the methods and the design up front. And in doing so, they might determine that there are actually some fatal wounds there, which means you couldn't realistically conduct an observational study to answer the question you're trying to answer. So the first thing is what's the question you're trying to answer? And we've seen a lot of self-inflicted wounds, to use Miguel's term, in a lot of the published studies we've looked at. And the time zero is a big issue, both for eligibility and for treatment assignment. Some of the published studies of hydroxychloroquine looked at time zero for follow-up as the day of admission to the hospital, but then treatment was started at different times after admission. So the propensity score matching was done at the time of admission, but after a few days, even after a day, two matched patients could have very different hospital courses, and their eligibility would have been different had that been assessed at the time the treatment was assigned. If you look at more chronic diseases, where there's a lot of interest in seeing if already marketed medicines could be used as, say, a second line for a chronic treatment, and perhaps and in situations where it's used off-label, we see, let me just say, it's hard to know when to match the treated patient to the untreated patient. Yes, treated patients are different from untreated patients. That's where the randomization issue comes in. But if you're looking for at a cohort of patients, all of whom have been treated with the same initial treatment and then progress, and some of whom receive a new treatment and some of whom receive nothing, how do you know that they meet the same eligibility criteria at the same time? What is that time zero for the untreated patient? And so these, I think, are some of the big challenges we have in using these kinds of data, not to mention the fact that observations are taken at routine intervals in clinical trials when they aren't in clinical practice, and more data may mean more severe, a more severe clinical situation, and thus confounding what we measure. So this isn't easy, but I think my panelists have presented, or my fellow panelists have presented a lot of good comments here. And I'm very much a believer in the emulation of clinical trials. And so I think we really do have to pay attention very, very carefully to the methods. So with that, thank you.
Great, thanks, Gerald. And that does seem like, as Gerald said, some consensus among the panel, this direction of emulation. And I might ask you all, if we got a question about it too, is emulation the right word, replication, et cetera? This approach based on the target trial model is something that is very valuable for expanding the effective use of real world evidence development. I'd like to go back to a couple of the issues that Adrian raised in his comments that came up in Gerald's as well, or Gerald, you touched on this. How do we, can we tell in advance? I know, Miguel, you laid out some criteria, but in advance, whether observational methods are going to be informative, and maybe just keep a little bit with the COVID-19 examples. You focused on tocilizumab benefit, and that's actually a case where the observational study preceded the randomized trial. But as Adrian and others have pointed out, there are, I don't know how many hydroxychloroquine observational studies or convalescent plasma observational studies that were not replicated in randomized controlled trials. Any comments you'd like to add, Miguel, or others, about how we can tell in advance about whether observational trials will be informative? And Miguel, did you predict these findings for the different observational studies in COVID?
Well, it is not possible to know for sure, but we can think about some red lights that we may want to use. In the case of COVID, just to stay here, if you think about the example I gave you of tocilizumab, we were looking at this data for a very long time, and we've reached the conclusion that we couldn't find any possible bias that would go in the direction that would explain the benefit of the drug. So it's not only, it is a two-stage process. First, you say, look, let's do everything that we can to eliminate these self-inflicted injuries, like the immortal time bias, and we have to make sure that we compare patients on the first day at the ICU with patients in the first day, and patients in the second day with patients in the second day. After doing all those things and having something that looks as close as possible to a randomized trial, except for the fact that treatment has not been randomly assigned, the only thing that is left now is the possibility of confounding. So the first step is getting rid of all the other things and make sure that the only major bias you can have is confounding. After you are at that point, then evaluate what is the magnitude, what is the expected magnitude, what is the direction of the confounding. In this particular case, it was impossible to think of how confounding could explain this going in the other direction. If you think about the other drugs or the other interventions that have been mentioned, in many of the observational studies, the first step was not done. There was no analysis of the observational data in such a way that it looks like a randomized trial, except for the randomized assignment. And there are some, of course, that very infamous examples of hydroxychloroquine. It also helps when you have a paper with 600 hospitals or more over the world and only 3 or 4 co-authors. That usually is a red light too.
Good point. Adrian?
Yeah, I guess, I mean, I kind of view COVID as maybe not the ideal example for us to go forward with in terms of emulation to expansion of information that can be actionable, in part because of COVID, and especially at the beginning of this, there's so much panic. Use anything no matter what, because of the issues that, the unmeasured confounding is really difficult. And so this is what I was talking about, the essential issue about, it's easy to have advanced hindsight. It's harder to have advanced foresight, especially in a pandemic setting. But I think we can do things going forward in terms of if you emulate phase 3 studies and then look to questions or ancillary to that to start paving the pathway for using observational methods for understanding different populations, their ancillary or populations that are harder to study. The other thing about the COVID experience here that at least one observation from all this is that because of all the different learnings in terms of just misunderstandings about the biology or the clinical practice, systems weren't really in place to even have other things would be important about measuring issues around COVID-19. How we measured things, had electronic health data even for COVID diagnosis was different in March compared to May compared to August. And so I think that's another issue that comes up.
Yeah, thanks, Adrian. Gerald, did you want to add something on this?
No, I think they've covered it pretty well.
Okay. Any other comments on this question? I guess I have a related follow-up. This came up in the Q&A chat. Said, well, and this definitely happened in COVID, right when the pandemic began. As Adrian said, we weren't set up as we'd ideally like to have been for getting randomized trials done fast, and still working on that problem. But there were a lot of compounds that were already approved for use. So there's already some significant safety and effectiveness data that might have been beneficial for COVID. So we talked about a few here. Is there another path forward here? That is, if we try to take the steps that Mael described and Adrian added to and do them right, can we use observational analyses to help guide us to where we might want to do a confirmatory or really a lead randomized clinical trial, given the limited clinical trial capacity that we have in many areas of unmet medical need, like COVID, like other conditions as well? Can these methods be used to help guide further clinical trial targeting?
I think that's something. I just think that, like, if you have 1,000 options, for example, so-called off the shelf, and as you noted, the trial capacity is like this, then you have to do something, especially rapidly. How do you narrow down from 1,000 or plus options to something that maybe a dozen or less? So that certainly is right.
I would just add in, COVID is an interesting example because we didn't know what was going on, but there's a lot of treatments that are already out in the market for supplemental approvals, things like that, that I think you'll hear about later on, where this RWE trials might provide that target trial. And that may be more promising as we think about expanding patient populations, expanding the outcomes, et cetera.
Any other comments? Well, let me now take this the other direction. And there are a number of questions, comments that have come up along these lines. Okay, let's assume we've got a couple of clinical trials done, perhaps enough to constitute substantial evidence for approval of a product as safe and effective, and at least some indication. Is it possible to use observational methods to extend that effort, maybe starting by, and this was Adrian, I think you raised this too, if you can start by some level of replication or duplication of the clinical trial, could you then have more confidence in using the same methods to address related issues, other outcomes, subpopulations, use in different clinical contexts than those studied in the randomized trial?
Sure. This is an idea that we've been discussing for a very long time. As Jennifer said, we call it benchmarking, and that's the whole point. You have a trial, and you know the results. You use the observational data to try to match those results. And this, as you were saying, Mark, can be used to extend to a different type of patients, to slightly different treatment strategies, or even more importantly, to just a longer follow-up. In some cases, you can do a relatively fast trial with 1 year follow-up, looking at at the biomarker as the outcome, and then extend. And once you meet that benchmark, then you can extend it to a clinical outcome over 5 or 10 years using observational data, if there is so much experience in the real world. The challenge here is to define what benchmarking means and when you are finding the same results as the trial. My colleague Isa Dahabre has been working on this for the last few years, and he's very actively developing these methods to really come up with a formal framework for benchmarking that is more satisfying than simply looking at the numbers and say, hey, they look kind of similar.
Other comments on this? Any particular areas that you'd highlight where this benchmarking approach, I get the fact that this is in development. I think we're going to talk more in later sessions about how you'd actually apply a benchmarking framework effectively, but any lead examples, promising areas where you all think this approach might be particularly relevant?
Yeah, I mean, there's some examples we're just thinking about, like, hey, there's a drug that showed, great benefits to risk in the outpatient setting. The question comes up, can you do it start in the inpatient? So in some cases, people have had to do clinical trials to prove that, and it largely can replicate, but takes time. And so then there are patients who miss those opportunities, for example. But you'd have to start to prove that you could benchmark that. Other things that for certain, like populations that have like a very small percentage of the clinical trial where there's inconveniently a subgroup that's on the wrong side of one. Is that the truth, or is it because there are 20 subgroups? And so how would you be able to understand that without having to another clinical trial for that subgroup? So those to me are concrete examples where it could be informative, and if structured the right way, it could even be included in information that's published.
Great. Thank you. We've had a couple of questions from people who are joining today about definitions. So duplication, replication, emulation. Anyone want to take that one on?
Well, I can say that we can only talk about duplication or replication for trials that have already been conducted, and we are trying to get there. Whereas the concept of emulation is of a target trial is broader because it also applies to target trials that have not been conducted, but they may be conducted in the future.
And there's a difference between duplicating the results in a given population in a study that's been done versus replicating that study, perhaps in a somewhat different population or somewhat different data set.
Yeah,
okay.
I mentioned this in the words matter. I think that outside of probably people on today's call, people might often expect a replication means that not only the primary outcome, but all of the secondary outcomes will be replicated. And I think that gets pretty dangerous as we think about, for example, in cardiovascular studies, a lot of the MACE endpoints include angina, which is really hard to measure in observational studies. And so I think as we talk about this, not only do we need to get clear, but we need to make sure that those outside of our conversations also are clear.
Thank you. I know we're running a little bit over. We built in a slight cushion. I do want to get in one more question that relates to, I think, covers several topics that have come up in the Q&A and discussion. So thank you all who are joining us today for participating in that. This gets back to the journal editor reactions that you mentioned earlier. It doesn't seem like, and I'm paraphrasing some of the questions here, that there are some well-established procedures that journals use or that reviewers, that people who are participating here could use if they were reviewing a study or designing a study for how exactly to apply the kind of framework that we've talked about here. Are there good resources available that sum this up? Is this still work that's kind of in process, or some combination of those?
I think that there are a lot of resources that are in place already for randomized trials, and many of them are easily applicable to a large extent to observational studies. Take CONSORT as an example. CONSORT was designed as a set of guidelines for randomized trials, but a lot of them could be applied to large follow-up studies from observational databases. The fact that it is not done, the fact that we don't require that observational cohorts are reported following say, a CONSORT flowchart that we require for all the trials, or the fact that we in many observational studies we also obtain a hazard ratio but never the absolute risks, the cumulative risks like we do in the trials. There is a lot of low-hanging fruit that just by taking what we already do for trials and applying to observational studies could get us a long way there.
Nandita, maybe a related kind of final question. You talked about how you're getting a lot of requests from your collaborators for conducting different types of observational analysis. Is there any place that you can steer them to help get to a more systematic and efficient approach to applying effective methods like the ones or potentially effective methods like the ones we've described today? You talked about Spiros, for example. Neil mentioned some resources have been developed for clinical trials. Anything you'd like to add? Oh, may have lost Nandita. Anything that the rest of you would like to add?
Sorry.
Okay, there we go.
Sorry.
Thank you.
The first thing I do is usually give them Miguel's paper to read from AJE, because most of my collaborators aren't familiar with this emulating the target trial idea. And I also just wanted to second what Miguel said in terms of reviewers of papers and journal editors about observational studies. And oftentimes they bring up this, there's some unmeasured confounding that you couldn't deal with. That's the major thing that they focus on. And then if we throw in a sensitivity analysis, they're fine with that. But, you know, the bigger issues oftentimes don't come out in terms of like the time zero and things like that. But yeah, I oftentimes now just start with giving them the Miguel's paper and kind of start the conversation there, which really helps, rather than the checklist, because I find the checklists like Spiros, they still kind of, they put in these details about what their plan is going to be without actually thinking about why they're doing that or what they're emulating.
Okay, great. Well, I want to thank all of you for a great discussion. Neil, thanks for the presentation, a framework that we're all clearly able to build on. So the one takeaway lesson is Neil's paper and the related steps that you all are developing to make it easier to follow this kind of target trial approach. And I guess the second thing is, if you don't remember anything else, time zero, immortal time bias. So thanks for everybody, for everyone in this panel for a great discussion. We're going to take a break now for about the next 13 minutes till 2:45. We'll start back up again with our next session then. Thank you all very much. All right, we are indeed ready to start. So welcome back. And I'd like to welcome you all to our next session, which will include presentations from trial replication projects. We're going to hear 3 presentations on preliminary results from ongoing trial replication efforts. And building on the last session, the goal here is to better understand the strengths and limitations of observational studies focused on questions about drug product effectiveness and regulatory decision-making using the kind of framework and ideas that you've heard about so far during our web event today. And I'd like to begin by introducing our speakers for this session. Sebastian Schneeweiss is a professor of medicine and epidemiology at Harvard Medical School and chief of the Division of Pharmacoepidemiology in the Department of Medicine at Brigham and Women's Hospital. He's going to present results from RCT Duplicate. Bill Crown is a distinguished research scientist at the Heller School at Brandeis University. He's going to present on Operand. You'll hear more about these replication efforts in their presentations. And Nilay Shah is chair of the Division of Healthcare Policy and Research in the Department of Health Sciences Research at Mayo Clinic. He's going to present on behalf of the Yale Mayo Clinic CIRCE project. So first we'll go to Sebastian.
Thank you very much, Mark. I think, Valerie, you want to pass the control over to me? Thank you very much. So thank you, Mark, indeed, for having me to speak this afternoon. We want to share the first findings and our first learnings from emulating randomized controlled trials with real-world evidence studies. I want to acknowledge Jessica Franklin and Shirley Wang. Together, we are the leadership team of this family of studies under this umbrella of RCT Duplicate. The study that I'm presenting today is entirely funded by the FDA under 2 contracts. You may want to pause the video stream in order to read my disclosures. At the end of last year, a paper was published in Circulation that summarizes the findings of the first 10 emulations of a total of 30 randomized trial emulations that we are conducting under this contract. And I'm building on this today. And today I show you the first 20. Here you may want to read the participants from the Harvard study team, the Edion team, our colleagues from the FDA who have always helped us throughout the process, and the many reviewers from FDA who have provided insights, our expert advisory panel. I want to point out that the authors are solely responsible for the presented findings today. It was mentioned already this afternoon that we see real-world evidence is based on clinical practice data as complementing real-world evidence from evidence derived from randomized controlled trials and expand on the evidence generated through randomized studies into different populations, into different treatment patterns, co-medication patterns, treatment strategies, different endpoints, different comparators. So it is not either randomized or real-world evidence, it is the complementation that they bring to each other. The key question that we are asking ourselves all afternoon already, can real-world evidence studies ever estimate causal treatment effects? And when we do associative studies, whether we are explicit about this or implicit, we are talking about treatment effects of what causal treatment is affecting, we draw conclusions thereof. Therefore, if we want to calibrate then our real-world findings against some sort of truth, where do we gain knowledge about the truth? And once we know this, of course, we can just look at the difference between those two treatment effect estimates and therefore conclude there is overlap or not. If we are not God, how can we ever know what the true treatment effect is in a given population? So we have several options here. One is relying on expert opinion. What is a causal relationship, what not? And that's probably not so good. We know a lot about statistical simulation studies. Even there, I think we can learn a lot from them, but still it's a synthetic universe that is generated. So it's not really what we are talking about here, real life data, clinical practice data. So the next best thing might just be that we compare against randomized controlled trials, assuming that a well-planned and well-executed randomized controlled trial is accepted as having a causal interpretation about the treatment effect in the population that we're interested in. So that is the possibility, and that is our key assumption when we do these emulation studies in order to calibrate whether real-world evidence studies can come to causal conclusions. Well, if real-world evidence cannot estimate causal treatment effects, what is the point of doing real-world evidence? We make recommendations for treating our patients with these studies. We need to be sure that it is the treatment that is causing the outcome and not the disease. When you engage in these types of projects, you realize very quickly that you are in between several fronts that are fighting each other. And one community, for example, is the community of real-world evidence proponents. And they say, well, real-world evidence studies answer different questions in RCTs, and therefore you should never expect the same findings. You should not compare. It may backfire, I heard, which translates to me, well, we can never really test validity of real-world evidence because we don't have an agreeable gold standard to test against. And that is deeply dissatisfying. And we can quote Karl Popper and many other philosophers of science that if you don't even have a testable hypothesis, what are we doing if the hypothesis evades testability? And another community is randomized trial proponents who might say real-world evidence studies have never been able to convincingly demonstrate that they have causal conclusions like randomized trial controls have, which translates for me to, well, the bar is set high, we're open to listen, but we doubt that real-world evidence will ever be trusted. What these emulation projects try to do, they try to get these two communities closer together with empirical data and in a more systematic way and identify where can we come to causal conclusions? Are there any red flags, as Miguel mentioned already, that might be raised when there might be an issue and maybe we are not coming close to causal conclusions? What we don't mean, we don't want to imply that all real-world evidence studies need to calibrate against an RCT. Well, that would, again, totally defeat the purpose of real-world evidence because it's meant to complement the RCT evidence, not to replicate real-world evidence, the RCT evidence. This is really a learning a project where we try to understand how well and how predictably can we come to causal conclusions. Now let's dive in and understand variability. Variability between RCTs, variability between real-world evidence studies is a good starting point here. And John Concato published some 20 years ago, a paper in the Journal of Medicine, where we identified 5 therapeutic areas. The first one here listed is the BCG vaccination against tuberculosis. The black dots are the RCTs that were identified in literature, and the open circles are the real-world evidence studies that were identified. And what you see is there is quite some variability from the left to the right, moving left to the right, between RCTs as well as between real-world evidence studies. There's overall some overlap between those two worlds, but there's also quite some variability amongst these studies that seem to study the same issue. So if we study to what extent real-world evidence studies can emulate RCTs, we need to consider that there's quite some variation between real-world evidence studies, and you will see this later in my presentation, the estimates between different data sources, for example. But you also need to recognize there's variability amongst RCTs. If we run 2 RCTs, we don't expect exactly the same numeric finding. Now, if we don't find the same finding in a single RCT, in a single real-world evidence study, several questions come immediately up. One is, well, what is the agreement, what we're agreeing to or not to? What is the variability between RCT findings? And does this one RCT that we have identified truly reflect the estimate in our population? Is there an emulation failure in this specific exercise? Did we really manage to mimic the population of the trial population as closely as possible? Do we observe the same treatment patterns? Did we manage to emulate the outcome measurement? Do we have the same follow-up duration, and so on? This is what we call emulation failure. And then the third option is, well, we have a bias here. There's residual confounding operating. We have differential surveillance. We have misclassification. We have time-related biases, and so on. And it is the latter category, the third category, that we are really interested in these emulation exercises. We're trying to answer the question in an optimally conducted study, as good as it gets in a practical sense, what is the bias that we still have to deal with? And we want to minimize emulation failure issues, and we also want to minimize the variability between RCTs in order to answer that question in a targeted way. Now, this is kind of illustrating kind of the relationship between how well you succeed in emulating the randomized controlled trial. It's the quality of the real-world evidence study and the closeness of the emulation. This is on the x-axis in blue here, and in green on the y-axis, you see kind of the expected agreement between the real-world evidence study and the randomized controlled trial. And of course, there's not a strictly mathematical relationship, but we sense the better we emulate the randomized trial, the higher quality data we have, the closer we are likely to get to the RCT findings. And this is a bunch of retrospective emulations like the Colcado study that I mentioned already, where the randomized trials and real evidence studies were identified in literature. Our project is about here. We have very targeted emulations. We identified a trial and then mimic the trial as closely as possible. And there are other exercises where you do post-hoc re-rating of your population towards the trial. We did not do that, or these double randomized study designs where we actually find almost exactly the same finding as the randomized controlled trial. Now, very narrowly, the FDA-funded project where we wanted to replicate 30 RCTs and predict 7 ongoing phase 4 RCTs, where we very narrowly wanted to learn whether if we had replaced an RCT that was submitted or should have been submitted to the FDA with a single real-world evidence study, if we replaced RCT with a single real-world evidence study, would we have come to the same regulatory decisions? It's a very narrow question. As you see already, the single real-world evidence study and single RCT is making the stakes quite high rather than splitting this. We wanted to develop a process of how to do these emulation studies and how to share the findings with a stakeholder like the FDA and allow the FDA to do sensitivity analyses on their own, like they're used to when they're getting randomized trials submitted. And we also, of course, in the longer run, not talking about this today very much, identify factors that get us closer to the RCT findings. Now, let's dive into the methodological framework that we used for these emulations. We limited ourselves to claims data, to US claims data that have enrollment files, that have patient level longitudinal information on visits, hospitalizations, pharmacy fills. In that, we have diagnostic information, procedure information, and very detailed information about the medications that were filled. Specifically, we're using the Optum Clinformatics claims database, the IBM MarketScan claims database, and Medicare fee-for-service claims data of beneficiaries 65 years and older. The RCTs were identified by trying to reach a mix of the regular type of regulatory submission, primary approvals, supplemental approvals, negative trials that never made it to the FDA or were rejected by the FDA, and several studies that were provided by colleagues from the FDA that were special interest in those studies. We wanted to have a mix of therapeutic areas. We wanted to have a mix of comparators, placebo comparators versus active comparators in the randomized trials. We wanted to have a mix of the hypothesis testing intention, superiority testing versus non-inferiority. In the trial selection strategy, very quickly we were focused on the fit for purpose of the claims data that we have to even be able to emulate the measurements that are necessary to emulate the randomized controlled trial. Is the outcome observable and measurable and decent specificity? Is the treatment observable? Key inclusion exclusion criteria observable? Can we mimic the study population therefore? And are the pre-exposure confounding factors observable in order then to overcome the lack of baseline randomization? The study design analytic strategy, well, we emulated a target trial. The target trial was given to us, so we don't have to think very long about this. It is described and we just emulate this. This leads us very naturally to the new user study design. We are trying to always find an active comparator in order to reduce potential confounding here. We emulated the inclusion exclusion criteria as best as possible, given the data that we had, the claims data that we had. We achieved or tried to achieve balance of pre-exposure patient characteristics by one-to-one propensity score matching on more than 100 pre-exposure covariates in all of these studies. We tried to use validated outcome definitions when available. When not, we tried to have as specific definitions as possible. we wanted to emulate the ITT analysis in the randomized control trials, where we often observe very, very good compliance, more than 90% compliance. So patients are on the treatment as assigned from the very beginning throughout their participation in the trial. And in the light of the suboptimal real-world adherence to medications, we used an on-treatment analysis to mimic the ITT analysis because we assumed that the compliance would be close to 100% in these trials that were submitted to the FDA. We have only one single predefined analysis. The task was to put all eggs into one basket and emulate the situation as if we had only one shot and would present that real-world evidence study then to the regulator. We have a single investigator team with our experiences, with our biases. We had clinical and methodological advisors who contributed very usefully. We had very few sensitivity analyses baked in this iteration. Of course, there are many follow-on sensitivity analysis later on. The process and feasibility, we were, as I mentioned already, trying to mimic the RCT population as closely as possible. On the right side, you see a typical document for this. And these documents are all posted on clinicaltrials.gov, where you see the inclusion-exclusion criteria. And in black, you see the criteria where we thought we can emulate them quite well. In blue, in dark blue, you see, well, they're sufficiently emulated with the claims data. Maybe not perfectly, but we thought it's sufficient. In the red criteria, we thought, well, it's really difficult to emulate. A little nugget here, a frequent exclusion criteria in randomized trial apparently is, well, if you don't survive the next 6 months, you cannot participate in a randomized trial. I don't know how a trial investigator is operationalizing that, and it's even harder for us with claims data to come up with a prospective rule who's going to die in the next 6 months and exclude them as of today. So, so there is difficulty here. Nevertheless, if we moved only forward if we thought the emulation is good enough and will not affect the primary point estimate of interest here, right? So there are lots of judgment calls in here, and you see the gory details in the posted documents. For each of the cohort studies that we use to emulate the randomized control trials, we have these design diagrams to illustrate the temporality. In the new user design, we do, in order to avoid adjustment for intermediates, we avoid immortal person time, we avoid depletion by susceptibles by this clear temporality and the onset of the follow-up and the end of the follow-up. Some of these diagrams are quite complicated in order to mimic the inclusion-exclusion criteria of the trial as closely as possible. We had 2 feasibility studies built in, and if we were not satisfied at any of these points, we would still stop the process and say, we're not going forward with the emulation of this randomized trial because we don't feel comfortable that we can ever come to something that is close to an emulation. Power is one of the checks. We expected a statistical power as good or as high as that of the randomized control trials, given the observed point estimate in the trial. Reminder that in the 30 replications, we knew already the results, right? So we used that as for our power calculation. And then we have a feasibility analysis about the balance in the pre-exposure variables that we can achieve. On the right side, you see these blue bars before any adjustment. You see quite some imbalances in some variables, and after propensity score matching, the distributions are much more balanced. And if you don't see any residual imbalances, we decided to go forward. Here you see, and that's already published in the CPT publication, the selection process of the trials for primary approval. Start out with almost 600 trials that we reviewed, and largely for measurement issues, most of the trials were excluded. In the end, we have 17 primary approval, 13 supplemental, some negative trials, and some FDA suggested trials. As you can see, this portfolio was actually more than 30, and we are still open to recruiting more trials because some of these trials still drop out in the feasibility analyses. This is the process that we have in the graphical depiction, moving from the left to the right, the data quality checks that lead to these many aborts where we said we cannot move forward. But then once you pass the feasibility analysis, and we still thought it's worthwhile doing the emulation, then we register the protocol before we do the associative study, do the analysis, then send the report, share the results with the FDA, and allow for feasibility and for sensitivity analysis, changing definitions in conjunction with the regulators by sharing the analytic environment and the access to the data, although the data were residing still with us. Here's some examples of why some randomized trials that were originally thought to be reasonable targets for the emulation dropped out. These are antiplatelets trials. The first two, they dropped out because we then realized, well, the treatments were largely given in the hospital, and with the claims that you don't have very good visibility into that. And the other three, they dropped out because the medication of interest was used so infrequently in these databases that we have available. Although they're very large, some of these medications simply don't have that much traction in the marketplace, and therefore we let them behind us. We use the Edion evidence platform as our software tool to implement the studies in a very transparent, very systematic way, following the typical analytic strategies, and we can then share the Edion evidence platform with the regulators in order to replicate these findings. This is the structure of the document that we are depositing on clinicaltrials.gov. 1 through 12 are really all activities that happen before the comparative analysis starts at step number 13, where we then run the analysis that was predefined and deposited at clinicaltrials.gov. Here's an example of the document that is deposited. There are hot links there that get you directly to the platform in order to replicate the findings, allowing the FDA to feel comfortable with the data and the type of analysis. We had predefined agreement assessments, where one assessment strategy is what we call regulatory agreement. It's very much a p-value based agreement rule, where if the p-value would lead you to the identical regulatory decision, then we call this regulatory agreement, or RA. Estimate agreement means that the point estimate of the real world evidence study is within the 95% confidence interval of the randomized controlled trial. You see this orange real world evidence study. The point estimate is within the 95% confidence interval of the trial. Therefore, we have estimate agreement. Here, that's not the case, and therefore we don't have estimate agreement. So these are binary decision agreement metrics. And then we, of course, we have the numeric difference between the estimates. We quantify this in the standardized difference, and if that is not statistically significant, then you would reach the SD criterion for standardized difference. These are 3 binary agreement statistics, and we will learn that this is not always optimal to have binary agreement statistics. We looked at the quality of the emulation, which is critical, the quality of the comparator emulation. We usually judge them to be good if there was an active comparator in the trial, moderate to poor if it was a placebo comparator. And we had to find alternatives to that. The endpoint emulation, good or moderate, whether the endpoint could be identified with high specificity. Now with that, I dive into the findings. The first 10 randomized trials, 8 of them were second-line antidiabetic medications, GLP-1 receptor agonists, SGLT2 inhibitors and DPP4 inhibitors. The 8th study, the Carolina trial, was actually a prediction of the findings of the trial, while the other 7 were replications. Like all the other studies that I'm showing you today are all replications, except for Carolina. We have 2 antiplatelet trials, Triton and Plato. I want to then point out our assessment of the comparator emulation among those 10. And we see that the first 7 trials had placebo comparators: Leader, Declare, Emparec, Canvas, Carmelina, Ticos, and Savor, Timmy. And what we did is, for the first 4, for example, the SGLT2s and GLP-1s, we used as an active comparator DPP4 inhibitors, because we know that DPP-4 inhibitors have no effect on the endpoint of interest, which is 3-point MACE, major adverse cardiovascular events. And therefore, yes, they do reduce blood sugar as an antidiabetic, but they're not affecting the outcome that we are studying, and therefore should emulate a placebo, right? So in with regard to this outcome, we have this next set of 3 here, where we have, you see the red marking here, where we emulated the placebo group for the DPP-4 inhibitors with sulfonylureas. And there was already a little bit of a concern that users of sulfonylureas are quite different from those using DPP-4 inhibitors, and we're worried about that. Here, the last 3, we have active comparators, glimepiride, clopidogrel, for the 2 antiplatelets, and we felt quite comfortable with these emulations of the comparator group. With regard to the outcome emulation, you see overall, we feel very comfortable with the 3-point MACE endpoint. In yellow, a bit moderate, we have the heart failure hospitalization and CV deaths. In these claims data in the United States, it's very difficult to get robust data on the cause of death. So we have all-cause mortality observed and not the cardiovascular specific mortality. And the angina is something that we also observe not very well. We catch a lot of rule-out anginas. We might be missing some true anginas as well. So it's non-specific, and this might lead to some issues. The randomized trials 11 through 20, the second batch, we have the first 3 being direct oral anticoagulants in patients with atrial fibrillation for the prevention of stroke, Aristotle, Reliant, Rocket AF. The next 3 is equally direct oral anticoagulants, but now for the treatment of DVT, Einstein DVT, Recover2, and Amplify. The number 17 is Paradigm HF for treatment of heart failure. We have antihypertensives, Transcend and On Target. And the last one of this first batch of 20 is Horizon, pivotal, which is for the treatment of osteoporosis. And you see again the markings here in green and yellow. Many of them are active comparators. Therefore, green with the heart failure Paradigm HF, enalapril was emulated by any ACE inhibitor in order to increase the number of patients in our study in order to reach statistical power. And in the Transcend group, we emulated a placebo with loops, calcium channel blockers, and thiazides, which was allowed as kind of the background treatment in the Transcend trial. So we felt reasonably comfortable, but not perfect. For Horizon pivotal zoledronic acid against placebo, we used raloxifene as an emulation of the placebo, also knowing that in Horizon about 30 to 40% of subjects were actually using raloxifene in the background. So, but still not completely perfect, but a reasonable approximation. With regard to the outcome, the first 3 trials, the DOACs in atrial fibrillation, we felt very comfortable to measure stroke. This is any stroke, that is the hemorrhagic stroke as well as the ischemic stroke lumped together, systemic emboli. We felt quite comfortable. With regard to the VTE assessment, we're working with diagnostic information. We don't have Doppler exams and things like that, so they could be better. And then, of course, again, we don't have cardiovascular specific deaths. With regard to the hip fracture, and the very last one, we measure hip fractures quite well, but we were concerned that we have a shorter follow-up because of the adherence in real life. Let's have a look at the distribution of the event rates across the first batch of 10. Again, I just highlight in color marking our quality assessment of the outcome emulation. Let's look at the 2 pink columns here, the event rates in the exposure group in the RCT and the event rate in exposure group in the real world evidence study. And then likewise in the light blue columns, you see in the comparator event rates between RCT and real world evidence. What you see overall is that the event rate is higher in the randomized controlled trial. These are enrichment strategies, and we forced very high specificity endpoint definition, therefore might have missed some true events. However, there are two exceptions, declare as well as TCOS, where we have higher event rates. And these are exactly TCOS, for example, where angina is our concern, that we have a more sensitive and less specific endpoint definition. And therefore, actually, we have higher event rates in the real world evidence study, 7.3 versus 4.1. The next batch of 10 randomized trials, where we see, again, in the first 3 trials, we see very similar event rates in the direct oral anticoagulants in patients with atrial fibrillation for stroke prevention. I mentioned already that we felt very comfortable in the outcome emulation for all strokes. The next set of 3, we found higher event rates in the real world evidence study because we had these less specific endpoints, and then the lower event rates in the real world evidence studies because we have the lower sensitivity endpoint definitions. This is typical Kaplan-Meier plot where we see the upper 2 lines are the experiences in the leader randomized controlled trial, where the black line is the placebo, blue is liraglutide, and then you see kind of shifted downwards in the incidence rate of stroke, the DPP-4 inhibitor, which we use to emulate the placebo, for the 3-point MACE endpoint, and then in red we see the liraglutide in the real world evidence. But you see the proportionality actually mimics quite nicely the randomized controlled trial. Before I go into the treatment effect estimates, we also identified tracer outcomes to calibrate our methodological approach, and a negative and positive tracer outcomes where we had strong prior knowledge about the effect size and the effect direction. And every green check mark here that you see in the very last column means that we reached our goal, that we could convince ourselves that we replicate these findings of these tracer endpoints, except for one outcome, the pneumonia hospitalization in the triton population, where we thought there should be no relationship, and actually we saw some preventative effect, which is not really explainable. Overall, when you look at the scatter plot, where you have the log hazard ratio of the RCTs versus the log hazard ratio of the real world evidence emulations, they're huddling around the diagonal. Ideally, they would all be exactly on the diagonal, these dark blue dots, and they aren't. Some are very close, some are further away. You see, for example, horizon down here, there's a bunch away. And that is exactly where we have the learning opportunity, what exactly would explain this. So I now go batch by batch here to explain the findings that we observed. So for example, the first trial, the leader trial, moderate comparator emulation, good endpoint emulation, the RCT finding 0.87 with the 95s here, the real world evidence result 0.82 with the 95 here, standardized difference. The type of test was non-inferiority, and here you have the 3 agreement statistics: regulatory agreement, estimate agreement, and standardized difference agreement. Overall, you see that we were quite happy with these emulations. Their point was quite similar. What you see and declare is that the trial showed 0.83, while the real evidence emulation showed a stronger effect size, right? To the extent even that the 0.69 was not even in the 95% confidence interval of the randomized trial, right? But from a regulatory perspective, that would still be coming to the same conclusion. But clearly, from a scientific perspective, that is interesting to observe and maybe dive deeper. The trials 5 through 8, these are you see over here on the right upper corner, this little cheat sheet here. These are the DPP-4 inhibitors. They are the first 3 against placebo, the 4th one against clemepiride, and we emulated against sulfonylureas. What you see here, these are all null findings in these trials, right? Null findings with regard to 3-point MACE. But when you look at the first 3 findings here, we would come to the conclusion, following the regulatory agreement logic, that the sulfonylureas, the DPP-4 inhibitors have a better endpoint, better finding than the sulfonylureas. And as such, we give ourselves asterisks here. We still, strictly speaking, we still reach non-inferiority, but of course also we seem to reach superiority, which the trial did not show. The Carolina trial, we actually reached this, the regulatory agreement as well, if you want to look at these binary agreement statistics. But I want to point out that when you compare Carolina against TCOS, right, the randomized trials were had the same point estimate, 0.98, 0.98, with somewhat comparable 95% confidence intervals. And look at the real-world evidence studies. The 0.91 that we have in Carolina is actually not that different from our TCOS estimate, 0.89. It's really quite similar. Just turns out that we have more information in the TCOS trial, and therefore the 95s are tighter than the 95s for Carolina, which spans the 1.0, and therefore we reach this kind of this binary metric of regulatory agreement. Right? You see already the issues when coming to simple conclusions that is often difficult to draw for any of these emulation exercises. The antiplatelets, prasugrel and ticagrelor, Triton and Plato. We were quite happy with the emulation. The RCT findings 0.81, 0.88. We have success in all these three binary agreement statistics. The point estimate is slightly attenuated towards the null. The Plato equally attenuated towards the null. Here, of course, then the 95 is spilling over the 1, and then according to the regulatory framework, it would no longer qualify for regulatory agreement because it would be considered a null finding, while the randomized trial showed the benefit here. I want to point out that it's not clear whether this is a bias or whether it is an emulation failure, because when you look at the Plato trial, it was, of course, a multinational trial where in the United States, actually, there was no superiority of ticagrelor, while the trial was entirely driven, the success of the trial was entirely driven by, I think, many of the European sites and non-US sites where there was a substantial benefit of ticagrelor. It is hypothesized that, and we know that in the United States, some patients used higher dose aspirin, the 325 milligrams versus the 100 that is frequently used in Europe. And the hypothesis is that maybe that has interfered with the effectiveness of the ticagrelor. I don't know, but the fact is that we use US databases, and if we compare ourselves to the US finding only, then the interpretation changes a little bit here, right? So that is the struggle with emulation exercises like these. Trials 11 through 13, Aristotle rely on Rocket AF with regard to stroke prevention in AF. The point estimates here, 0.79, 0.65, actually quite similar overall. We were quite happy with the findings. Although, again, strictly speaking, 0.65, the point estimate here in the real world evidence, did not fit into the 95 of the randomized trial. So we could not give us this binary estimate agreement statistic here, or agreement. Randomized trials 14, 15, and 16. These are the direct oral anticoagulants in DVT treatment. Good compared to emulation, moderate endpoint emulation of the DVT. Look at the point estimates here, 0.6875. This is one instance where we have more information in the real world evidence. The point estimate is more towards null, it's attenuated, but the 95% confidence intervals are more narrow because we have much larger populations here, while the 0.68 of Einstein DVT is actually crossing the null with this upper 95 confidence interval. That's, strictly speaking, we did not have full regulatory agreement here. Again, that is kind of a limitation of a binary agreement statistic. Overall, we felt quite good about all these emulations, 1.08, 1.10, 0.84, 0.776. Paradigm HF, where the randomized trial had a solid 20% reduction in heart failure hospitalization and CV mortality. We could not reproduce that in our real world evidence study. We are still struggling and trying to find out what happened in our emulation. We had some concern at the beginning already. We did not have New York Heart Association class in order to classify the severity of the heart failure. We didn't have ejection fraction. We are looking at the database specific findings, and you see market scan has the point estimate is 0.85, while in the other two databases we don't see any effect here. We also stratifying by age. Is that our older population than a trial population, maybe we don't see the effectiveness as strongly there. In a randomized trial, there's some hint for that. I don't want to overinterpret a randomized trial subgroup analysis clearly, but this is also opportunity for me to point out that although we have exactly the same age inclusion criteria, right, within that bracket that is allowable in a randomized trial, we still have different distributions in the age. Often towards older age in our population. So although we mimic the design, we emulate design as much as we can, we might still not have the same age distribution. Number 18 and 19, transcend on target. Transcend worked out quite nicely so far, 0.92, 0.88. We were quite happy with that. Although we had, again, more information, tighter confidence intervals than the RCT. On target, it's a null finding, it's a non-inferiority trial, and we still need to try to find out what exactly went on here that we cannot emulate this. We were quite comfortable and hopeful and that we will be able to mimic that trial, particularly since we had already done an emulation study earlier and published that with a slightly different population, earlier data and things like that. We need to find out. So these are the learning opportunities here. Clearly on target falls into that. Horizon, our osteoporosis trials on nitric acid, and the RCT findings 0.59, we had 0.75. While we hit the agreement statistics, right, it's still puzzling us why we don't see a stronger treatment effect here. So we looked at the Kaplan-Meier diagrams from the randomized trials on the left side here. And what you see in a randomized controlled trials for the 36 months follow-up, you see this 0.59, right? That's the RCT finding. If you look only the first 18 months, you see 0.75. So there is some indication of time-varying treatment effects, that the treatment effects actually becoming larger as you're longer on the treatment. And of course, in our real-world evidence emulation, we truncated follow-up at 18 months because we had very few patients actually coming back for a second injection after a year for solitonic acid. So we had basically no adherence after 18 months or after 12 months, really. But if you look just at the first 18 months, the point estimate's identical, but that's luck or fluke, whatever you want to call it. But clearly we come to the same conclusion in the first 18 months. So you see that there is an emulation mismatch. But if we focus the emulation, then we have the calibration success. I did not fiddle around with the numbers up here. The numbers are the pre-specified analysis. We want to be totally honest to our word here on the pre-specified analysis, but it is interesting, of course, then to explain. Very briefly, the confounding adjustment, how much is changing here. You see now on the very right column, the unadjusted estimate from the real world evidence. I haven't shown this before. And the second to last is the estimate that you have seen already after one-to-one propensity score matching. And you see in the red boxes where there was substantial change from the unadjusted to the adjusted, but you also see examples where there was very little movement, where the active comparator design was taking care of most of the observable confounding that might have been here. And similarly, in the second batch of 10 studies, there are quite a few examples where actually intensive confounding adjustment of observable factors did not change very much in the point estimate, which again highlights the importance of the study design. And when you look at across the 20 trial emulations, the database specific point estimates, want to point out two examples. Overall, they're quite aligned, but the two examples where there is some discordance between the three different data sources, like here for Carolina and for Recover2. But every time we see this discordance, we see also this very wide 95% confidence interval. So the estimate is really just bouncing around. In conclusion, what we think are the first learnings here, with data that are fit for purpose and proper design and analysis, non-randomized real-world evidence studies usually come to the same conclusion about a drug's treatment effect as randomized trials do. Fit for purpose data, proper study and design. These initial findings of RCT Duplicate program indicate circumstances when real-world evidence may offer causal insights in situations where RCT data is either not available or cannot be quickly or feasibly generated. The complementary nature of RCTs and real-world evidence. Some learnings we need to take into account in any of these emulation exercises, the inherent uncertainty in any single randomized control trial. It may not really be reflective of the causal treatment effect in that study population. Similarly, one wouldn't likely take only one primary result as a single RCT in isolation. You would do sensitivity analysis in order to explore this. Our challenge was to compare only one estimate over here and one estimate in the real-world evidence. A single binary success metric will not do justice. I think you learned this through my presentation. You need multiple metrics. In any emulation, despite best efforts, there will remain differences particularly in the population, in the study population, in other measurements of the outcome, and in the drug use characteristics. So that is certainly our observation that in order for these emulation exercises to go well, you need very good measurements of population, the comparator, and the outcome. If the data are fit for purpose in study design choices, this is much more important, it seems to us, than the nitty-gritty of the analytic strategy, which often doesn't have that much of an impact. We remain concerned about three emulations, the paradigm HF, on target, and saver TIMI. We need to find out, and this is the learning opportunity here clearly, why do we get such divergent finding? Are these biases operating? Are these emulation failures? What exactly is going on here? And my last slide here, what do we make out of this? If we get more and more of these emulation exercises, there will be some generalizable conclusions. I think many of them we know already today, and they will be solidified by these exercises. But the most important outcome may very well be that we have a repository of well-documented studies that illustrate the agreement between RCTs and real-world evidence. And then in specific situations when the real-world evidence study is explicitly designed to answer the same question as the RCT, right? So these are important data points, reference points, right? They can be sorted by therapeutic area, by the data source, is it claims, is EHR registry data, the type of comparator, the type of outcome. With those reference points, then when we do real-world evidence studies, we can then say, okay, well, I operate in the same therapeutic area, I use the same data source, I use a similar comparator. Can I now, by making the analogy, like in case law, we built this case by case, can I make an analogy back to this well-documented emulation exercise, and from that draw insights and eventually draw more increased confidence that, yes, I will come close to any causal conclusions with this real-world evidence study. And with that, I give back to Mark.
Great. Thanks very much, Sebastian, covering an enormous amount of work on this Replicate RCT project. Congratulations to you and the rest of the collaborators for doing it. We're going to go on to two related presentations now. I did want to generate a lot of questions, many of them about specific studies, Sebastian, that don't go off yet. I've got one question for you. A lot of questions about specific studies. Don't have time for those right now. Hopefully wait to the next discussion. But a number related to the reliance on claims data in a lot of this work, with questions about whether that might account for some of the mismatches that you described, and just some general questions about, hey, you don't have hemoglobin A1C results or a lot of other potentially relevant clinical information here, yet you still were able to proceed quite far, maybe because these data are very well understood. So can you just add a few comments about the implications, the focus on claims here, and what that means for other data that might be more clinically enriched, but maybe less well understood or complete?
So we have this self-imposed limitation that we decided upfront. We work with claims data for this exercise here. We have a follow-on project where we go into EHR data. But we selected the randomized trials in a way where we felt comfortable that we can measure key variables. clearly we still lack the clinical granularity for some. So HbA1c was mentioned. I mentioned New York Heart Association class, ejection fraction, the heart failure trials, and things like that. There is never really enough information that you would like to have in order to fully mimic the randomized trial, and not with claims data. And I would argue with electronic health records, often you won't have that either. And you get another bag of problems with EHR data potentially. I think the future might very well be in the combination of claims data with electronic health record information to have the best of both worlds in order to get as complete data fit for purpose for the study question that you're interested in.
Great. Thanks very much. And thanks again for the presentation. So next I'm going to go right to Bill Crown.
Oh, thanks very much, Mark. Thanks to you and the organizers for this session for including me. What I'm going to do is talk about another trial emulation project called Operand, which is really a study that I'll focus on the sort of the complementarity of where Operand fits relative to RCT Duplicate. And Valerie, if you could give me control. Let's see. Thank you very much. Operand is attempting to study additional sources in variability of treatment effects from these observational studies through comparisons with RCTs, looking at not only inclusion, exclusion criteria, study design, ability to emulate the study design characteristics of the real trial, but also the decision-making of the different researchers attempting to emulate the same trials, sensitivity to data analysis, the statistical methods that were used, and then also looking at the heterogeneity of treatment effects as the inclusion exclusion of the trials, criteria of the trials were relaxed. So we started with the emulation activity, and then we looked at the broader real world population. We had one of the trials was the same as was in the RCT Duplicate results, the Rocket AF trial. We had picked that particular one because, like Sebastian pointed out, we felt that the stroke and the stroke outcomes would be particularly good for being able to emulate with claims data. But then the second trial that we looked at was the LEAD-2 diabetes trial. As you were just pointing out, Mark, this had as its endpoint hemoglobin A1C changes. So for this one, we picked it as a type of study which would normally not be possible in claims data, but would require electronic medical record data to conduct it. The data we used were in the Optum Labs data warehouse. At the time that the studies started, I was the chief scientific officer of Optum Labs. And the Optum Labs data warehouse is a very large data set. It's about 130 million lives of claims data. And it has 85 million lives of electronic medical record data, some of which overlap with the claims data. So the idea was to sort of bring those two different types of data together and look at analysis with the claims data alone and the claims data plus the electronic medical record data. We initially restricted to the inclusion and exclusion criteria of the trial and the design of the trial as closely as possible. And then we looked at the broader patient populations. Each of the two different research teams, I'll get into more detail on this in a moment, were had only the initial pivotal publications to go by. And the idea was, how would you emulate the trial if you had the information that was in the original pivotal publications? They were given a prescribed set of methods. So we asked them to use, single equation regression modeling or generalized linear models. We asked them to use propensity score matching. We asked them to use inverse probability weights. And then we also asked them to use research methods of their own choosing in addition to those. But we were especially interested in Operand on the how the research decision-making of the teams influenced the approach that they took in the analysis. So there's sort of a bounded guardrails here, because they already had the trial, the actual trial to guide them in terms of how they set up the study. But even so, there were some very interesting results that came out around that decision-making. The Operand was co-led by myself and Barbara Beerer at MRCT Brigham and Women's Hospital, and had a number of pharmaceutical sponsors who provided unrestricted grant money for this process. We put together a technical expert panel that helped with the design of the study, and then also we went through a request for proposals to the Optum Labs Academic Research Collaboratory to submit proposals, and Brown University and Harvard Pilgrim Healthcare Institute were selected as the two research teams. So you've heard about the rocket atrial fibrillation trial already, so I won't go into more detail on that. The LEAD-2 diabetes trial was really focused on efficacy and safety of adding liraglutide to metformin against placebo or glimepiride. As we've heard, comparing against placebo is difficult. So most of the comparisons here were against glimepiride for the analysis in Operand. Like RCT Duplicate, we had two of the three measures for agreement measures. We used the regulatory agreement measure, and we used the statistical agreement measure. Also, like the RCT Duplicate, both research teams pre-published their research protocols on EU PASS registry before they were giving any access to the data. So I'm going to give you only high-level results from Operand. We're just about to submit the findings for peer review, but I want to highlight a couple of major things. First was the issues of the cohort identification and the characteristics of the samples that were identified by the two different research teams for the two trials, and how those compared versus how they compared to the trial characteristics. And for both research teams, the sizes of the cohorts and the characteristics that they identified for the rocket atrial fibrillation trial were very similar. So they really had using the rocket atrial fibrillation pivotal publication, they applied similar inclusion-exclusion criteria, and since they were using the same data, naturally they identified very similar samples. One group chose to apply slightly more restrictive criteria than the other, but the end result was minimal. There were substantial differences for the LEAD-2 trial, and this is very interesting because the difference in the sample size came about largely due to how the research teams addressed availability of follow-up hemoglobin A1C. So there was, in the data, there were large numbers of patients with baseline hemoglobin A1Cs, but it was much rarer to find the follow-up hemoglobin A1C levels that were required in the trial. And this goes back to the interpretation of how the teams interpreted what the exercise was. Even though they both understood that it had to do with emulating the actual trial, in one case, it was a matter of designing the target trial to answer the same question as the real trial. And the second group interpreted that more narrowly to mean I want to follow the same inclusion-exclusion criteria as the actual trial. And this led to big differences in sample size because the actual trial required follow-up hemoglobin A1C levels in order to measure change in hemoglobin A1C levels. And that introduced a very large missing data problem for the follow-up hemoglobin A1C. The other research team dealt with that as a censoring issue and used inverse probability weights to adjust for the missing data. Interestingly, the comparisons of the baseline characteristics of the samples that were identified by both groups were very similar. So the missingness didn't seem to be informative in the sense that the samples were very similar to one another. And in both cases, the samples that were identified in the observational data were more similar to one another than the comparisons to the randomized trials. And much as Sebastian was pointing out, in the observational data, I would say that for Rocket AF, the observational cohorts were less clinically severe, I would say. The CHADS 2 scores were skewed towards the lower levels. They were less likely to have had previous congestive heart failure. They were less likely to have had previous stroke or systemic embolism. So in the case of the LEAD-2 trial, the differences were less noticeable with the RCT, except that in the observational data, there was a much higher prior use of sulfonylureas, and then also there were differences in racial composition of the observational data relative to the trials, with the observational data being having a much higher percentage of non-white patients than in RCTs. And again, when you think about the fact of the RCTs being conducted globally, the trials in the United States being the US-based data for the observational data, those sort of differences may reflect differences in populations geographically around the world. In terms of the treatment effect estimates, both of the teams generated treatment effect estimates that were very similar to their respective trials. And that was true in terms of using both the regulatory and the statistical comparison methods. So across both those methods, they were similar. There was very little variation in the estimates by statistical methods. There was variation, but it was not large. And I want to come back to echoing the points that were made earlier around the importance of these design issues and such as the defining of time zero versus the statistical methods. These are the issues around bias are often not so much around the statistical methods and control for confounders as they are for much more fundamental design issues. Interestingly, for both of these trials, there was very little variation in the treatment effect estimates when the inclusion-exclusion criteria were loosened. These results obviously are not generalizable to all other disease states or different clinical interventions, because other areas might well lead to much more heterogeneity in treatment effects in routine clinical practice than in the narrower populations that were studied in randomized trials. But certainly in the case of Rocket AF and the LEAD2 emulation exercises, the broader real world evidence population looked very similar in terms of treatment effects to the randomized trial. I had presented these results earlier. This is just to give you a sense of the amount of variability in the treatment effect estimates about a year ago. These are preliminary results for the Rocket atrial fibrillation study. And up at the top is the actual trial estimate here. And then I've interwoven results from using different methods by the two different research teams. And I think up here in the beginning, these are unadjusted for anything, just the inclusion-exclusion criteria. And then broadly, as you go down the road, the rows, the methods become more sophisticated, dealing with things like censoring and so forth. But there isn't a huge amount of variability in these estimates, as you can see. And the results were similar for the final results and for the LEAD2 diabetes estimates. So what have we learned from these emulation efforts? I think when we're doing these, the design of a target trial for when we have an actual trial to attempt to emulate, we can still have results that may differ in important ways in design of that target trial from the actual trial. So if the desire is to answer the same research question as the actual trial, the way that one might do that might differ. You might decide not to have follow-up hemoglobin A1C levels for everybody in your cohort, but rather deal with that with censoring, for example. But we do have this growing literature now on these emulation efforts that have shown that it's often possible to arrive at very similar treatment effect estimates with observational data. And I think as Sebastian was pointing out, we can learn probably the most from areas where things disagree. And one can imagine, for example, oncology trials where we don't have cancer stage in these observational data sets, and we generally don't have it in the electronic medical record data either. Without cancer stage, one might imagine it's very difficult to balance for disease severity in observational data analyses, and that's just a simple example. I think we often find that in these emulations that the sample characteristics of our observational data look somewhat or sometimes quite different from those in the randomized trials. that's worrisome, but not necessarily fatal. We of course need to go through these exercises of attempting to balance the cohorts as best we can for anything that we can observe in the data. And there may be unobservable things that are correlated both with the treatment selection in the observational data and the outcomes, which could introduce bias. But just because we are missing, just because the characteristics are different in the observational data and the randomized trial doesn't mean that we can't control for them with through the multivariate analysis. That's an empirical question, whether it's possible to control for those. And then I think finally, I'll conclude with, as the data and methods continue to improve, we continue to work on methods to improve. There's a lot of interest now in, for example, building dynamic treatment regimes into these sorts of analyses. I think the ability to be able to improve the reliability of causal inference with observational data should continue to improve. With that, I'll turn it back to you, Mark.
Okay, thanks very much, Bill. And next we'll go right to Nilay Shah.
Great. Thanks so much for inviting us to present here at this meeting. Let me just make sure I have access. Yep. So I will be presenting on behalf of our relatively large group that's been working as part of the Yale Mayo Clinic Center for Excellence in Regulatory Science and Innovation, funded by the FDA, and the work we've undertaken in terms of emulating trials. And you can see this work was supported both by FDA, but also I'll talk about one of the trial replications that was funded by NHLBI. So in addition to our internal team, we also work closely with the FDA real world evidence team, a lot of the same folks that Sebastian mentioned earlier in his presentation as well. So just to give you an overview and some things to think about as we go through the presentation here. One is something that has come up is thinking about data sources and what are the ideal data sources for looking at a variety of questions, especially in the context of emulating clinical trials. The population representativeness, so even when we emulate trials, what are the potential differences in population? Third issue is the impact of real world practice versus clinical trial design. So how you might design a clinical trial may not be how the real world practice works. And you'll see in a couple of the examples that we have quite different approaches as to what happens in real world compared to clinical trials. And how do we account for that as we emulate the trials? We'll discuss a little bit about approaches to address selection. And then as we think about data sources, as well as a variety of other issues, I think the critical piece also is how do we get endpoints that we can feel comfortable from within the real world data and what that might look like in the future as well. We'll discuss 3 trials primarily here and dig into them a little bit with specific issues that we'll discuss. So one, we'll discuss the PRONOUNCE trial and the GRADE trial. Those 2 trials were conducted with the FDA in partnership, and the CABANA trial, which was emulated separately as part of an NHLBI award. And the key consideration in each of these 3 trials is we emulated trials to predict both the population and the results of the trial prior to publication of the results from the trial. So the whole idea was we don't know the answers, and especially as you'll see with PRONOUNCE and GRADE, those trials are still underway. The results are not released, so you'll see the early results that we're seeing from the real world data, but how they play out with the trials, we are yet to know. CABANA, we were able to emulate the trial prior to the completion of the trials, and we'll compare the results from both those pieces. So just to give you an overview of the PRONOUNCE trial. So the PRONOUNCE trial was a safety trial, randomly allocated 900 participants with advanced stage prostate cancer and cardiovascular disease to either daguerreolex or leuprolide. And the main goal was to look at adverse cardiovascular events. The trial, again, as I mentioned, is active, not recruiting, but the results are currently not available. So when we use the real world data, we were able to identify about 8,000 patients who would be eligible for the PRONOUNCE trial after applying both the inclusion and exclusion criteria. We removed potential crossover in this, although I'll get into that in more detail in just a moment. And we identified about 1,300 patients on daguerreolex and 6,700 patients on leuprolide. And what you'll see here, again, this is not to get into the detail, but a point that the panelists earlier brought up is, when we start with a population from real world data, we oftentimes start with a very large population. Here in this case, you can see we started out with 103,000 individuals receiving either of the drugs. But once you apply all the inclusion, exclusion, all the different criteria, you end up with a much smaller sample. So it's still oftentimes is still larger than what you would see in a clinical trial. We identified crossover in 336 days in 20 leuprolide patients, but the key was in the daguerreolex arm. We saw 810 patients that actually did receive leuprolide. And as we had the urologist on our team sort of pointed out, this makes clinical sense due to the longer dosing interval for leuprolide. So this will commonly happen in real world practice. So again, going back to this issue of how do we sort of marry what we are seeing in real world practice with what happens, what's proposed in the clinical trial. So we also conducted 3 analyses, but also a number of other subgroup analyses that I won't present here. We conducted an intention to treat as our primary analysis, but we also conducted secondary analyses, which focused on censoring of patients and dropping any patients that crossed over between drugs. And we use the typical one-to-one matching. In this case, propensity score matching looked at standardized differences of 0.1 or less for the 2 groups, and a Cox proportional hazards regression was used. And what you can see here overall in the intention to treat analysis, we don't see any difference in major adverse cardiovascular events. But when we drop the crossover groups, so specifically either the daguerreolex that crossed over to leuprolide or vice versa, we actually do see a difference in major adverse cardiovascular events. So we're trying to understand that a little bit better of why that might be different. I think our urologist colleague who sort of led the study, he did not expect that difference. So I think we are looking into that, but you can see the difference of when you make those assumptions of intention to treat versus when we drop the crossover and look at sort of the pure group in that context. The second study I'll talk about a little bit is the GRADE trial. And the GRADE trial is the glycemia reduction approaches in diabetes. It's a comparative effectiveness study. It's a 36-center pragmatic unmasked parallel treatment group trial. It randomly allocated 5,000 adults with type 2 diabetes with an A1C of 6.8 to 8.5%. And I'll come back to that in a moment. That becomes important as we are trying to emulate this trial. And patients were allocated to one of 4 drugs: glimepiride, sulfonylurea, sitagliptin, a DPP-4 inhibitor, liraglutide, a GLP-1 receptor agonist, and glargine, a basal analog insulin. And the study was conceived in 2008, and the recruitment was between 2013 to 2017. And we are hoping to have some results mid to late this year. But the key piece for those that follow the diabetes literature is you'll notice the SGLT2 inhibitors, which are not part of this trial, even though they're becoming more and more commonplace in diabetes treatment. And the time to primary metabolic failure of the assigned group was the initial A1C greater than 7%, subsequently confirmed at the next visit while being treated at maximum tolerable doses of metformin in the assigned treatment. And the A1C was measured quarterly for 4 to 7 years. And one of the things when we replicate this information, again, the richness of the data we were able to use, and I didn't mention that earlier, but it's Bill did a great job describing the Optum Labs data warehouse, and we use the same data source for replicating each of the trials here. And we were able to leverage for both of these studies, both PRONOUNCE and for GRADE, linked data to electronic health records, which again increases the richness of the data. And even though it increases and provides the clinical information, the anticipated deviations from GRADE, you can see we cannot require confirmatory A1C like the trial requires, just because that typically doesn't happen in real world practice. And we also anticipate random and non-random variation of when the A1C testing and results are available. So that really is different than trials. As we try to emulate it with a clinical endpoint, there are pretty significant variations in that context. And as you can see, again, the main point here is the sample size across these 4 groups that I wanted everyone to sort of look at. And specifically with the glargine group, there were only 251 individuals who met the trial criteria. And one of the challenges is that glargine is typically not prescribed for those with A1C of 6.5 to 8.8 as the trial specified. So it's not surprising that we did not see more patients in this group. So again, the difference here between what the real world practice does versus what the clinical trial specified becomes quite different. And so what we ended up doing is we actually ended up dropping the glargine arm due to the small sample size. And even after dropping the glargine arm, the groups were still not balanced due to liraglutide group being still relatively small, partially potentially driven by the cost of the drug, as well as oftentimes it's prescribed as a 3rd-line agent and not a 2nd-line agent. So we performed a variety of approaches to sort of, weight the different cohorts. We used a generalized boosted logistic model and used stabilized weights, which again, in this next slide, as you'll see, that gets us a little bit closer and a little bit better balance with the stabilized weights. But with the other approaches, we saw a fair amount of variation across the 3 groups. The results of this trial are not complete. The emulation will be completing it shortly, and the trial results won't be available until later this year. And lastly, I wanted to talk a little bit about one of the trials where we've completed this approach with the Cabana trial. And the Cabana trial was one of the largest trial in electrophysiology. It compared ablation to medication therapy for atrial fibrillation, and it randomized 2,204 patients. And in parallel, we also initiated a NIH-funded study that was conducted in parallel and completed prior to the data lock for Cabana. And as we look at the Cabana findings, the intention to treat analysis showed a hazard ratio of 0.86. However, the per protocol as well as the treatment received analysis had slightly different results. The per protocol analysis showed a hazard ratio of 0.73, and the treatment received was 0.67. And so it led to the question, even though the trial was completed, really, there was quite a bit of discussion in the community, in the electrophysiology community. How do you interpret the results of this trial? And in parallel, we had been conducting this work, sort of looking at 3 big questions. Do trial participants represent patients in everyday practice? So when we apply the criteria to the real world data, do the patient characteristics match? Are they similar to the clinical trial patients? Can observational data help interpret the trial findings? How do those data compare? And what is the treatment effect in the excluded population? So patients who are receiving these treatments but would not have met trial criteria. I won't discuss number 3 in this context. We'll primarily focus on the first 2 questions here. So do trial participants represent patients in everyday practice? So here you can see overall, when we apply the inclusion-exclusion criteria, about 74% of the patients are eligible, would be eligible to participate in the trial. 4% failed to meet inclusion criteria and 22% were excluded. The one thing that was interesting as we looked at even the eligible population compared to the trial population, the eligible population from the real world data tended to be older, more likely to be female, and a much greater number of minority patients. So it was interesting, even though you look at the same criteria, the distribution of characteristics, as I think both Sebastian and Bill have pointed out, tend to be different oftentimes compared to the clinical trial population. And, I won't get much into the propensity score analysis, but again, we used overlap weighting and used 90 baseline characteristics to conduct that analysis here. So here you can see the Cabana per protocol analysis, the hazard ratio was 0.73. When we used the Optum Labs trial eligible patients, it was 0.70. And very similar to what we expected or what we saw in the randomized trial. The one reason the per protocol analysis as well as the intent to treat analyses were different was because there was a 30% crossover in the trial. So as opposed to the crossover that we described earlier with the pronounced trial, where the crossover was between, in the real world population, here the crossover was in the trial. And so we tried to emulate that as we conducted the real world analysis. And so the crossover we were able to emulate after we saw the trial results. Prior to that, we conducted the analysis as just regular intention to treat analysis. And what we observed overall was the Cabana intention to treat analysis. The hazard ratio was 0.86. The Optum Labs real world data analysis was 0.85. So very similar, but I think as Sebastian saw in some of the trials he presented as well, much tighter confidence interval here. The Cabana treatment received, as we can see, the hazard ratio was 0.67, 0.73, and a similar analysis in the real world data was 0.70. So as we think about some of the key considerations of real world data for emulating clinical trials, one is sort of thinking about the data sources. The information on medication fills and clinical data together really add a lot. If we use electronic health record data, oftentimes the medication fill information is not available. And with the claims data, you oftentimes don't have the clinical information. So I think, as been mentioned a couple of times earlier, the ideal scenario is to integrate claims and electronic health record data that will also allow us to have better endpoints or greater confidence in the endpoints that are used. A second issue, at least in the future, that we hope will happen is that we'll be able to use the text data from electronic health records to use natural language processing and other approaches to, again, get better endpoints as well as covariates that can be used for these types of analyses. The population representativeness, even when emulating trials in real world data, this may be different. And to what extent that impacts the results that we'd see in real world data, we have to, I think, understand better through continuing to do more of these types of exercises. The third issue that I mentioned, the real world practice versus clinical trial design, as you can see a number of examples that we present in the 3 trials, there's a fair amount of difference of how clinical trials are designed relative to real world practice, especially when we have active comparator arms. And that's an important consideration whenever we think about where real world data can sort of meet the needs of replicating clinical trials. Approaches to address selection. I think in all the cases, we've at least used multiple approaches and conducted sensitivity analyses. This is an important issue to at least make sure that you see consistent results across different statistical approaches. And then finally, the real world results and clinical trial results will not always align. And I think Sebastian addressed that really well in the first presentation, but I think we'll have to sort of think about on a case-by-case basis, what does that mean? And how do we better understand that and learn from that when those results don't align? In some cases, it may be okay. In other cases, it may be a fundamental issue. Design of the study, for example, that was presented by Miguel earlier, the time zero not being defined appropriately and so forth. So with that, I think that was my last slide. Thanks again for inviting us to present here.
Great, Nilay, thank you very much for the presentation. And those of you who are following on the agenda can see we've gone a little bit into what had been scheduled as kind of an extra break time. So we are going to go ahead and keep going and finish a few minutes early, I think, just to stay on schedule for everyone. It's a great presentations and obviously a lot of material packed in. So we're going to turn to discussion just now. I know we've had a lot of questions and comments come in already. We're going to try to get to some of those. First, I'd like to hear from two additional panelists to provide some perspectives on this session of reactions to all of the replication results that have been presented today. So first, we're going to hear from Joe Ross, who's professor of medicine and public health at the Yale School of Medicine, who worked with Nilay Shah on the Yale Mayo CIRCE project that Nilay just described. And then Bob Temple, who's Deputy Center Director for Clinical Science for CDER at FDA and a senior advisor in the immediate office of the Office of New Drugs. So first want to hear from each of them, and then we'll go back to talk to some of the project leads about some of the common themes and issues coming out of the projects. So Joe, over to you first.
Thanks, Mark. And I'll keep my comments pretty short, obviously, because there's a lot to hear from the others on there's a lot of questions in the box that I think will be also interesting to cover. So thanks to Nilay for presenting on behalf of our team, and for thank you for inviting me to be a part of this panel. I think that what these three sessions, and I'm sorry that I missed the morning one, but I'm sure Miguel's also addressed this, is that there's obviously a lot of promise here. There's a lot of opportunity for using these larger databases to emulate these trials and to come to better certainty around the safety and effectiveness of these medical products using these data. To me, though, one of the things that I think is most critical is not just the granularity of the data and the endpoints that we are able to derive from it, but also the way to use it, I think, is in terms of sort of complementing trials and complementing and multiple different analyses. Because one of the challenges, per se, of course, is when even with a great database like Optum or the large Medicare claims data, when we're trying to better understand safety, effectiveness, what to expect when these trials finally do result out, or what to do when we can't conduct a trial, either it's too soon or for whatever reason. It's a population that's difficult to recruit. We want to be confident that the results we're getting from these observational data are robust. And so, doing multiple sets of sort of trial emulations, doing multiple different data sources, ideally across multiple different countries even, I think will bring us greater confidence as we're waiting on the trial, or when a trial cannot be done. The other thing is just, we just can't lose sight of the limitations of these data. There's a lot that we can learn from them, but they don't have a lot of endpoints and aspects that are really important in clinical care. We don't get symptom burden, we don't get functional status. There are things that we can only learn by directly going to the patients and asking them questions. They just don't appear in claims, or even in a lot of electronic health records currently in the structured data elements outside of lab results, and maybe not even sometimes imaging results. Sure, one day we may be able to glean this information from notes and charts, but there's we can only do what are in the data. And so they're just not to say that these are going to be great and perfect, but there's still a way to go, but they offer a lot. So I'll stop there, and I look forward to hearing some questions.
Great, thanks very much, Joe. And next I'll go to Bob.
Okay, I missed the early session, the earliest of the sessions, because my computer fell apart. I think the attempt to replicate all these, or duplicate them, is very exciting. It seems obvious you would, if you could, use data already collected to discover things, but you have to know that it works out. And I think even the materials we've seen are already making some suggestions. For example, if you look at basically underlying coronary artery disease, we have a pretty good idea of what the risk factors are, and we have a pretty good idea of what the endpoints are, at least for heart attack and stroke. And as long as we don't use something more complicated like angina events or something like that, it's not surprising to me that they show up pretty well, because we know the risk factors and the endpoints are pretty good. I'm also somewhat impressed that heart failure is more difficult, and I'm not surprised by that, because while some of the endpoints might be okay, they're a little more variable. Why was the person hospitalized? Was it the heart failure, or was it something else? So I'm not entirely surprised that it comes out a little different. it'll be very interesting also to see how osteoporosis works. I'm sure we'll pick up the limb fractures pretty well, but I'm much less optimistic about picking up various back events, which are much more subjective and things like that. So I'm very excited about the prospect of what's going on. I think it's very important to try to duplicate each one of these controlled trials more than once, and to look within the various the various databases that we have. That's helpful. I didn't see much discussion of how they differed in their outcomes, but that's a possible way to examine duplication. But what we really need to know in the long run is where these things actually conceivably could be a substitute for a randomized trial for one reason or another, and where they can't. I guess the one other thing that I'm not sure about, maybe people who've been speaking about it can, is how to estimate time zero when the control group is a no treatment group. I get it where you're comparing two different drugs, that seems easy, but how to do it for a placebo-controlled trial doesn't seem as clear at all. And when you compare it with a drug that doesn't have any effect, you don't really know that they were started for the same reasons. And that seems a very important concern because results change over time and things like that. But I just find the whole discussion very excited, want to see many more of these. And in the long run, we'll try to get to see how reliable this is. It does seem very important to go to areas other than the ones already discussed. We have a lot of cardiac events, coronary artery disease events. In some ways, those are the easy ones because those are because the risks and, as I said, endpoints are very well established. It's going to be more interesting to see about the more subjective areas, and heart failure is one of those, and I find that exciting too. So let me stop there.
Great. Thanks very much, Bob. Glad you got your computer back up and running. So maybe we could start off the discussion with Bob's last comment or question about how to estimate T0 for a placebo population. I think, Sebastian, you certainly covered a number of placebo trials where you sort of substituted an alternative treatment start, but that could lead to some differences in results. Would appreciate any thoughts you or others have about how to do this and what impact you might expect relative to efforts to replicate a placebo-controlled trial.
Clearly, as Bob already mentioned, we feel much more comfortable if there's an active comparator where we have a clear start date, we compare news of A versus news of B, and we feel much more comfortable. In the absence of that, it is tricky. You need very detailed disease status information in order to really find similar patients that are the same disease state, and some get treated, others don't get treated. And even then you ask yourself, what's the reason why they didn't get treated in routine care? So we have sensitivity analysis in the making where we emulate the placebo users by, we call them then non-users, but I'm not too optimistic about this with the data granularity that we have in claims data, so we feel much more comfortable. The question that you have to ask yourself, I understand from a regulatory perspective, where you want to have a placebo group. From a clinical perspective, right? I would love to hear from Joe as well and from Nilay. Is that really the question that we want to answer, right? When we write clinical guidelines, usually there are alternatives around, and it might actually be for a clinical guideline writing committee is much more helpful to compare A versus B rather than A versus placebo, right? I understand the regulatory position, but there are two different sides here to this end.
But there's still a concern about whether the stimulus to starting is the same for both treatments. The placebo issue still worries you. Oh, I should note, by the way, just to make it clear, when there's an event that leads to the start of treatment, like post-infarction treatments, then it's easy. You know what time zero is. It's when you're treating prophylactically that you have a problem.
Very much agree. So, Bob, it's also, we see this in osteoporosis studies. There's a fracture, which is kind of the index event, and some people get treated, others don't, and that makes it easier. I totally agree. An exception point.
One possibility, I think this is it's a question of whether or not this would be empirically possible or not, but in the electronic medical record data, you typically see the doctor's orders, but many times the patient never goes in and fills the prescription. So it would be interesting to see if one could create a matched group of patients that start on a given medication, and another match to a group that was prescribed the same medication but never filled it, and then follow them forward. And that might give you a placebo.
Although they might be very different populations.
That's very true, right.
Mm-hmm. Before we go on to some of the other topics, let me just ask the other presenters on any additional themes that you'd like to highlight in terms of similarities, differences based on what you've heard across this diverse set of replication studies. Any other take-home points for now?
I'll just make one quick comment. Although I think all of us have made the comment that statistical methods are clearly secondary to design methods, there have been some simulation results that have suggested that targeted maximum likelihood estimation methods in particular seem to outperform traditional methods from EPI or the econometrics literature. And I'll just sort of put that as something to for further work, because it may be that some of the machine learning approaches eventually may help us to generate, they may be helpful in the robustness of the treatment effect estimates that we generate.
Thanks. Any other comments? Joe?
Yeah, Mark, I guess.
Sorry, go ahead. Yeah.
Oh, sorry, Mark. I think you called me. I'll just say one more thing, just for the audience sake, and obviously, we're spending a lot of time thinking about how to use this type of approach for pharmaceuticals and biologics, and how to think about observational evidence in this space. And it's far more difficult in the medical device space, but just as critical and just as important. We're just much farther away from being able to do this type of work in general with data sources, given the lack of a UDI embedded within the CMS or other claims data.
Good point. Sebastian, were you going to add something?
Yeah, sure. I also wanted to highlight study transparency, right? And there are several comments in the Q&A there. Because as a reviewer of this real world evidence, you need to understand fairly quickly what exactly was done and then make a judgment about the potential validity. And in randomized trials, luckily, it is actually often very straightforward what was done, and you have fairly clear quality metrics of the data generation process and all that. And I think we need to do better on our real world evidence side, right? And there are software tools for that, of data provenance, the transparency of all the manipulation of the data from the generation by the treating physician to the analytic data set, be very transparent about the methodology and all that. And we can do that. It's very much, we can learn a lot from randomized trials there, right? So I think there's a lot of room for improvement there.
Yeah, and on that point, several of you mentioned, or you all implemented some steps like this, sort of pre-registering clinicaltrials.gov, advanced specification methods, et cetera. But picking up, there were a couple of questions about the importance of blinding, and certainly pre-specification helps with some of that. But unlike people who are doing the clinical trials, at least for the first time, you're not blinded to the results. And there's some questions along the lines of, well, do you all see any potential design issues around kind of playing to results that you may know are there? I don't see any easy way to blind investigators to widely reported published studies. So,
Mark, we're very, very, very worried about this. Not when you're trying to match a controlled trial. That's maybe a different question. But when you're trying to use it as the basic data. And we're working on writing guidance on this at this very moment, actually. But we will want to know that no one has looked at the results. You obviously have to look at the data to know how to do your trial. But we don't want a preliminary look at the results to shape the way the selection criteria are enumerated and to see who's included and who's not. One possible way to do it is to look at a prior database, form your conclusions, blah, 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 and then do the new study in a wholly different database. That's probably okay.
Right.
This is a very...
I agree with Bob here. Clearly, there is this concern that people peak and then design their study by looking at the point estimate and what they don't like, they discard and things like that. There are ways to make this more transparent. I thought, Bob, you were asking, because I think you were the one mentioning this, well, couldn't we just predict randomized trials that are currently ongoing where we don't know the finding? And of course, as you know, that's exactly what we do in our second contract. And you saw Carolina already published where we were successful in predicting but I should say, and this is a very personal experience really from the RCT Duplicate, even when you know the result of the trial already, and as an epidemiologist, you have to put forward one single study design with one single analytic strategy. It feels weird as an epidemiologist because we're used to always do a range of analyses to understand this activity. And I tell you, maybe just because I'm such an optimistic person, I always thought we were doing splendidly with these emulations, and you saw the results. Well, there were some studies where we did not, and I certainly did not foresee that. I thought we will do fine, right?
Well, we have to figure out the reasons.
Absolutely.
Some work and some don't. That's why this is exciting.
Yep.
The one area I just wanted to briefly bring up that we didn't discuss today, but there's been a lot of work going on with sort of distributed approaches to replicating trials, specifically through Odyssey. And what are the challenges when you're bringing various different data sources that are housed in different, in different settings and managed differently, and not a single group is looking at all that data? What's the implications of that? I think that's something that'll be important to consider, because Odyssey is worldwide. It has a lot larger sample, but what are the potential trade-offs you're making in that context?
Well, it's a good point, Nile. The studies reported here did a lot to try to, now the data came from different sources, multiple centers, et cetera, multiple EMRs or claim sources, but a lot of work curating those data sets beforehand to have more confidence about what's present in the data. I expect the same kind of issue comes up when you're trying to set up randomized trials at multiple centers. We've seen some examples of those in large-scale multinational platform trials, where there may be some differences. Do you or other, maybe Nile, if you could start by answering your own question, how are we doing in terms of putting together different data sources?
Yeah, and the Odyssey group has done a bunch of work in this space. I think having worked, I think primarily a lot of our work has been with the Optum Labs data, knowing all the sort of issues we find with the data when we drill in and are able to go upstream where the data are generated and understanding those issues. That brings up at least a pause when there's a variety of different data sources that are used that may be from many different organizations across the world and managed potentially differently. But I think there's a lot of interest as well in leveraging those data sources. So I think there's work to be done, at least I feel, in that space.
In a controlled trial done in various regions, you might expect that the populations would differ in risk factors and things like that, so that the number of events would be different. But because you're randomizing, you sort of still expect that the effect that you see, the hazard ratio, is going to be similar. In observational data, now you have to be nervous because the difference in the populations between the two treatments now can affect the effect sizes, not going to affect the difference between treatments as well as the effect size. So there's much more reason to be nervous.
Any other comments on this topic?
I'd like to make a couple of comments.
Please, Mayo.
That we have covered. One is that I think Bob has mentioned the problem of selective reporting, which is, I think, one very serious problem of observational studies that we don't have now in randomized trials as much as we used to. This is, by the way, this is also a problem in other experimental studies like lab studies with mice and rats, and this is not only about observational studies. But this is a huge problem in science, because if we are seeing only what the investigators were to show us out of the hundreds of things that they tried, it would be something that we cannot trust. And this was solved, or kind of solved, for randomized trial by mandatory registration when journals got serious about this. It has been proposed to solve this by mandatory registration for observational studies. I doubt that that is going to work, because most people working with observational studies have direct access to the data, so they can check. And why should they waste 2 hours for a registration when they can spend those 2 hours looking at the data, see what is promising, quotes promising, and then also only do a registration of what is promising. So I think that for observational studies, we need to look for other ways, because acting as the police, I don't know how the FDA or anyone can police this. I don't think that that's going to work. You can have built-in controls in the computer systems, so that in every organization working with large databases, the log of everything that has been done with the data is made public after that. That's one possibility. I don't think is beyond what can be done now. Another possibility, much easier, is data sharing. If all the data that is being used is shared, then anyone can check. At least some people can be, other investigators who go through an IRB can check. And just knowing that other people are going to look at the same data at the code that you have used has a deterrent effect. We see that in many other examples. So that is something that we haven't discussed here much, but besides confounding, the selective reporting of results from observational studies is a big problem. And the emulation of the target trial helps with that, because once you start from designing a randomized trial before you even look at your observational data, the protocol of a target trial is imposing a set of constraints of what you can do with the data. And that is at least a first check on what we're doing. I also wanted to say something about the placebo control that also Bob was mentioning, which is, I think it is very important. Look, there are some questions for which we need a placebo. And if we could use observational data to answer those questions, that would imply that we don't need the placebo in the first place, because there is no placebo in the observational data. So I don't think observational studies are going to replace trials. In fact, we need trials, and that is especially true for the placebo-controlled double-blind trials that cannot be emulated with observational data. We need trials as benchmarks, and then we can work with those benchmarks in the future with observational data. But we need something to start with, and that's why I think that for this question of observational studies versus randomized trials is misguided. Rather, it should be something like observational studies and randomized trials, because we need the trials as benchmarks, and we need the observational studies to build upon those benchmarks, and maybe to design better trials in the future too.
Great. Thanks.
I have one comment about placebo. I mean, the placebo is done to prevent bias. A placebo-controlled trial is a no treatment control with administration of a placebo to avoid bias. In an observational study where you don't really know that there's any comparison being intended, I'm not sure I'm that worried about the absence of a placebo control. You have a perfectly good no treatment if you can figure out what time zero is.
And here is where I think it is very important to think about what is the goal of a target trial emulation using observational data, and is to answer a question about what works. The question that can be answered is, treatment versus no treatment, or treatment versus an alternative treatment. It cannot be treatment versus placebo. That is not the question that we can try to answer.
No, that's right, but you don't need to. The placebo is there to blind you to the no treatment control. It's a placebo control is a no treatment control, but with control of your bias.
Right.
So in this setting, I'm not so worried about bias anymore.
I want to move on to another topic that came up in a number of questions in one form or another, and you all have touched on this. So most of the results that we've talked about today are from cardiovascular drug trials. Obviously, there are a lot of other therapies out there. And Joe, you mentioned some of the challenges in studying devices, not being able to identify the particular device in many observational data sets being a starter problem. Bob, you mentioned some of the, I don't know if the word softer is the right word to use, but outcomes that aren't quite as objective as a myocardial infarction or something like that. There are a number of questions about how applicable these methods are in other areas, though. So oncology, there's a question about use in rare diseases, where actually FDA does have a pretty good tradition of relying on so-called real-world data methods because of the difficulty of doing even primary clinical trials in some of those settings. Could you all comment a bit about extending some of the results that we've seen here to other disease areas, and what are the factors that you should be looking for in considering the fitness for purpose of these methods for replicating and extending or enhancing trials in other fields?
Look, I mean, if this is, to state the obvious, this is a measurement problem. It's not a methods problem. It's entirely a measurement problem, right? And clearly, the selection of the trials that we considered was because what are the endpoints that are well observable in claims data? In the next batch of trials, we have COPD trials, for example, where some of the endpoints are observable, but we stay away from many, many very interesting endpoints because there's no hope that we can ever observe them in a reasonable way in claims data. Now, with real-world data that are generated by the routine operation of the healthcare system, you always have the physician in between. First of all, the patient needs to go to the physician with some ailment. Physician needs to diagnose something. Physician needs to order tests. And if that's not done, there is no observation recorded. This makes them all very different from randomized trials or investigator design studies as primary data collection, where you go out there and actively collect all that information. In my mind, it will always be a limitation. And we will do better in some areas where the recording is more complete for whatever reason than in other areas. And we need to learn that. I think we need to understand that. And if you think about oncology, where it's a fairly controlled environment with very steady visits, usually when the disease is active. Even there, I think in the randomized trials, it seems to me, and I'm not an oncologist, there is difficulty with determining progression-free survival with reasonable reproducibility. And of course, that it will be extremely difficult with just observing what we see in electronic health record database. We have to look for alternative markers of endpoint, time to next treatment, things like that, and validate them, right? So we need to work with the reality of the data and see how far we can go and what alternative approaches.
But a lot depends on the endpoint and the size of the effect. I mean, single arm studies in oncology that are examining response rate are perfectly credible because responses don't occur by themselves ever, or almost never. So those are very reliable. If you have something that actually cures some people, you don't even need a control group because you know the response is zero. So in many of these rare diseases, the responses are sort of like that. You're basically reversing something that couldn't possibly change. It's when the differences between things are more subtle that you get hard. So response rate may be easy, but time to progression, if it's only 2 months difference or 3 months difference, you're probably going to need a control. It really depends on the size of the effect.
Other comments on this topic or extensions you'd like to see to other areas?
One thing I would just quickly mention is I think the ability to look beyond the carefully selected populations that are studied in randomized trials to patient populations that are treated in routine medical care, and particularly the effect of medical comorbidities and especially mental health comorbidities in terms of medical events that you can measure in these kinds of data, like hospitalizations and emergency room use and so forth. And all the questions that we have in the healthcare system around value, we're very focused obviously on regulatory questions here today, but these same issues apply to questions around assessing the value of treatments, the cost versus the clinical response of patients to different therapies. So I think that's a place where there's potential value to extend these trial emulation kinds of activities to other types of endpoints.
I just want to note that we are working hard to decrease what I like to call stupid exclusions from controlled trials too. But this is a way to get to the same end. Many of the exclusions by age, other disease are inappropriate because the very thing you want to find out is how these drugs work in a population that has other things. So we're trying hard to discourage that. I don't think we're there yet.
Yeah, this gets to another theme and a number of the questions that came up related to kind of going beyond the replication that's been the focus of today's discussion. We're going to get into some of this tomorrow, but to Bob's point, a number of questions relate to, well, what about extending these methods to other populations or to endpoints that can't easily be tracked over time in clinical trials, longer term outcomes, things like that. What about integrating additional data sources? So I know the focus has been on replication today, but I'd like maybe each of you to comment a little bit about suggestions you have for where we go from here. In particular, if you think about the FDA's framework for reviewing the totality of evidence for regulatory decisions, that obviously hinges a lot on well-designed controlled trials, generally randomized trials, but that's certainly not exclusive. And we've talked about a number of examples already where FDA relies on or regulatory decisions that go beyond that. Looking ahead, what's most important in the further development of these rigorous real-world evidence methods to be able to get to that point where you see more of a integrated approach involving both clinical trials and observational analyses that go beyond it? And maybe Bob, I'll save you for a little bit further down in this commentary so you can maybe react a little bit, but Miguel, maybe I can start with you.
I think I like to hear what Bob has to say if I can, because we have.
Okay, well maybe we'll follow do the other way. Let's start with Bob and then get reactions from the rest of the panelists.
Look, these are a variety of what I like to call hysterical controls, and that's that's one of the 5 kinds of historically of adequate and well-controlled studies that we identify in regulations and have done so since 1970. So it's possible these can be credible, but the big test is when they are. For the most part, actually, historical controls are credible when the effect sizes are large. What's interesting about some of the things that are being done here is that they're looking at, cardiovascular outcome trials where the effect sizes are anything but huge. And the question for us is going to be when and if these become credible and reliable enough to be used. So these attempts to duplicate the results are pretty exciting. And you can already sort of see that there may be some areas where it's more plausible than others, and we need to learn about those. So I wouldn't take that off the table. I think these are not impossible, but they obviously have to have a high level of credibility. We already use, so-called historical or external controls to see if the cancer shrinks. That's a no-brainer. But have you reduced the chance of dying from a heart attack? Well, we have some way to go, but it's an area of interest.
So thanks, Bob. And now Miguel, back to you. And then I'd like to go at least briefly through our other panelists. And this will be kind of our wrap-up comment for the session too.
I think that the use of external controls in this setting also needs to, we can use it if we keep in mind that the two arms of a trial have to have the same level of monitoring, the same intensity of observation, etc. So if we are going to use external controls from observational data, we cannot have an experimental arm that is that of a phase 3 trial. It should be the experimental arm of a pragmatic trial. And that's something to keep in mind, that we cannot mix the controls from a phase 4 trial, very large simple trials with the active arm of a phase 3 trial, and that applies to this same comparison. Having said that, with that in mind, I think that there is a very promising use of these studies to rule out treatments that should not be for which we don't have to invest a lot of effort in trials, because only with the observational data we can show that there is no way that this is going to work. Or the opposite, to find subsets of patients that are the most promising for this trial, for these treatments to work, and then that can be confirmed in trials.
Thanks, Miguel. Sebastian, maybe go to you next.
Sure. So setting aside the obvious use case that Bob mentioned already, when the effect sizes are gigantic or there's simply no alternative treatment available and the disease progression is very linear downwards and all that. Setting that aside, and also the external control arms, and also setting aside obvious use cases where you have very interesting active comparators in order to inform clinical practice, not as a regulator. When you zoom in a regulator, what always pops to my mind is supplemental indications, where you understand the molecule quite well already. You have often 2 phase 3 randomized trials in order to establish the efficacy. You've learned something about the safety. The medication's out there in the market, otherwise you couldn't do real world evidence, right? So one thought experiment would be, well, with the data out there, can you now first replicate the original indication, right? And if you feel comfortable there, well, now maybe we can go to another endpoint where you want to expand the label to, where we can go and we can expand the population. Can we expand into pediatric populations? Do we always want to do the pediatric randomized trials, or can we do this now with real world evidence because we have kind of calibrated against the trials that we have? So I see this in this very fruitful intersection where we have trials and we go beyond the trials.
Mm-hmm. Great. Bill, I see you nodding. Maybe I'll go to you next.
No, I was thinking the same thing as Sebastian. The other thought I had was sort of the value of conducting randomized trials in large healthcare settings. So the idea of going into a major health insurer, randomly selecting patients to be randomized, and then having alongside them an observational cohort that you could run right side by side a randomized population with an observational one. And I think we'd learn a lot in those sorts of situations where we would have a really, really similar base population that we're drawing from to investigate a whole series of different sorts of design issues.
Yeah, that would be great. And the VA can do those easily. So they could.
and maybe other health systems or coordinated care programs too. Nilay?
Yeah, no, I'll just add to what Bill said. I mean, there's different ways as we start thinking about more and more digital platforms and digital approaches coming online. Are there ways to not just use retrospective data, but prospectively recruit patients, especially where endpoints are not easily available in existing data? So are there ways to use digital approaches to both collect patient-reported data, collect other types of data that could be done more at scale rather than having, where randomization may be more difficult? There may be approaches to do that as sort of potential future areas. Like you said, there's very limited replication in terms of clinical areas. So as we get into other clinical areas, that might be an approach worth considering for the future.
Great. Thanks, Nilay. Joe?
Yeah, well, I'll just pick up on a couple of these things because as I see it, it's sort of where the front, sort of what's next in this area of space. And so, some of the things that Nilay and I are doing together in collaboration with FDA and others, is this sort of hybrid approach where we are randomizing patients at the point of care, but aggregating their electronic health record data prospectively, and sending out quick surveys and pulling in other information like digital device data, like wearables and whatnot. And that allows you to kind of create this sort of hybrid trial based within real world data. Other endpoints can still be collected, like satisfaction and other proms, but also you get the hard endpoints of coming back to, when coming back to a clinical system, or, and being admitted to the hospital or having a heart attack or whatever that is. You really are able to get the best of both worlds. Obviously, it's not quite as easy as working with an already existing database, but it allows you to do the randomized trial with a broader way, still reducing the burden and still leveraging real world data. The other thing I think that just important to think about is, the sort of frontier on how we think about data is what it's going to enable, right? Sebastian is right. It's the data limitations, not method limitations. And so, sometimes we do work within our using our health system data because there's increased granularity. That has its own, limitations in terms of follow-up and how long you're going to be able to see somebody coming back. But like in the emulations that Nilay and I have done as part of our work with FDA, we're leveraging lab data, right? Which is different than, we're already down to a subpopulation because it's not just the big claims, but it does allow you to get a little bit further and do a little bit more. What does it mean to have the PSA when you're trying to replicate the pronounced trial around, prostate cancer risk and cardiovascular safety? So, we have to be thinking about how to enable our structured data sources, our EPICs and other EHRs, our claims data to enable us to do better work in this space, because that's the major limitation.
Great, thank you. And Bob, any quick final thought? I appreciate your comments about the promise of this direction of work and also FDA's openness to engage on finding ways to build on it.
Well, obviously, I like the discussion of having these data serve as ways to make randomized trials more efficient. I mean, that part's to my brain, that's the easy part. We know we like that. And, the COVID experience has made us much more conscious of the potential for doing trials at a distance without having everybody come to a clinic because nobody wants to come to clinic. So I think that part is the part everybody likes. Whether and when observational data alone, not randomized, can become credible enough to be used is what, in part, we've been talking about. And I think there are some possibilities, and those need to be explored and understood better.
Great. Well, and that's, I think, a good point to leave off for today. I want to thank all of our panelists and presenters, not only for their very insightful and wide-ranging presentations and discussions today, but also for all the work in this whole area of study replication that brought us to today. So Bob left us with some questions about with this being a work in progress. I just want to remind everyone that this is day one of our two-day workshop. So we'll look forward to welcoming hopefully all of you back tomorrow for day two. We're going to start at 1 p.m. Eastern time. We're going to build on today's discussion about what the results from the replication project can tell us about conducting observational studies more generally and about fitting it into the work and hopefully opportunities for more efficient and effective learning from randomized studies to really improve that totality of evidence on medical products. So thank you all for joining us today. We'll begin tomorrow at 1 p.m. Eastern. You'll be able to access the meeting through the same link that we used today. And so in the meantime, have a good afternoon and evening.